Okay. Uh, can you hear my voice clearly?
Yes, Bu Maya.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. My name is Maya. Is your moderator for today. And with a good pleasure, I welcome you to parallel session 1 that would focus on the lignocellulose. Before we start the session, I would like to remind you that we will love to deliver an interactive session between you and the speaker. Therefore, for the presenter, we would love to invite you to turn your camera on when we start introducing you, playing your video presentation, and also at the beginning of the Q&A session. And for the participant, we also welcome you to do so. But if you are signed to turn your camera on, we would like to ask you to raise hand by click the raise hand button on your screen. And during this session, we recommend you to use any listening devices. And since this is an online forum, so you surely might enjoy this session with your colleagues at work and also with your family at home. Ladies and gentlemen, in this session, we will have two great invited speakers. We'll have Dr. Muhammad Zaki Hasan from University Technology Malaysia. Is Dr. Hasan joining? Okay, welcome and thank you, Dr. Hasan, for joining this symposium. And also we would like to welcome Dr. Munil El Akabi from Mohammed VI Polytechnic University, Morocco. Hello, Dr. Munil El Akabi. Okay, good morning. I think it's early morning, right? It's 6 or maybe 7 hours earlier behind the Indonesian local time. So thank you for joining with us. Ladies and gentlemen, before we start from the first speaker, I would like to read the short bio of the first presenter, Dr. Muhammad Zaki Hasan. So Ms. Rizka, could you please share the slide? Okay, I would like to read the short bio of the first presenter. This is Dr. Muhammad Zaki Hasan. He is an associate professor at the Razak Faculty of Technology and Informatics, University Technology Malaysia from Kuala Lumpur. Currently, he is head of the Vehicle Engineering and Safety Research Group, and also coordinate a team of researchers working on the diverse projects concerning advanced material, composite structure,
recording in progress,
and also polymer science. Now they have interest research focus on the additive manufacturing and also biomedical application. Okay, ladies and gentlemen, with the excellent background research and also many publication, I would like to start by playing the video record from Dr. Muhammad Zaki Hasan. So the IT, please, could you share or play the video?
Assalamualaikum and good morning everyone. Firstly, I would like to thank to you for giving me this opportunity, and it's my pleasure to introduce myself. My name is Muhammad Zaki bin Hasan from Razak Faculty of Technology and Informatics, University Technology Malaysia, Kuala Lumpur. A little bit brief of introduction about UTM Razak. The Razak Faculty of Technology and Informatics was established in 1992, which started out as one of the UTM Center of Excellence known as Business and Advanced Technology Center, or BATC, under an initiative conceived from the Prime Minister at that time, Tun Dr. Mahathir Mohamad. In May 2010, in line with the transformation of UTM Kuala Lumpur campus, this BATC was upgraded to faculty level and changed its name to UTM Razak School of Engineering and Advanced Technology. Again, on 1st of July 2018, the UTM Razak School of Engineering and Advanced Technology, Advanced Informatics School, and Perdana School of Science, Technology and Innovation Policy are being merged to form a new faculty that is known as Razak Faculty of Technology and Informatics, or RFTI. The merger was part of the UTM Synergy 4.0 transformation. Over the year, these three separate entities have developed strong linkages with a large number of industry and organizations, both locally and internationally, engaging in executive programs, short courses, professional discourses, community services, and innovation research. This community-like and industry-driven focus are also reflected in the content which are injected into all their undergrad and postgraduate programs. And now it has a solid team of academic staff comprising of more than 90% PhD holder from various disciplines. Its strongest asset, backed by its dedicated support staff. RFTI will work toward excellence and distinction. Today, I would like to talk about structural and properties of pineapple leaf fiber gel. As we know, the emergence of the development of bio-based material has extensively stimulated consideration among others. In Malaysia, the pineapple are mostly planted in state of Johor, Sarawak, Sabah, Kedah, Selangor, Penang, and also Kelantan. Among the varieties of pineapples planted in Malaysia are Morris Gajah, Josephine Yankee, Gendol and 36, and MD2. However, only two local varieties, namely Josephine and N36, has been successfully exported, mainly to Singapore and United Arab Emirates, because this variety have a long shelf life. And currently, the MD2 variety, which originated from Hawaii, is also planted and marketed in local as well as for international markets. In Malaysia, pineapple are grown in area around 10,800 hectares with an estimate production of 270,000 metric tons in 2015. The industry brings in over 600 million to the country's economy, according to the Malaysian Pineapple Industry Board. But harvesting pineapple results in tons of biomass waste such as pineapple leaf, core, peel, crown, and stem. Here, UMP researchers have developed and turned pineapple waste like bromeliad can also be found in the stem, core, peel, and leaf of the pineapple into usable items such as baby bottle, liquid cleanser, meat tendericide, tenderizer, and oyster mushroom production blocks. Also, Prof. Dr. Muhammad Tariq Hamdan from UP and University Putra Malaysia created drone parts as a sustainable method to reuse pineapple waste. Natural fiber cost low, reduced in density when compared to glass fibers. In terms of the strength of natural fibers, it is not as great as glass. The specific properties are also comparable. As we can see from the table of properties of the natural fiber composites, glass fibers composite significantly of outstanding mechanical properties and performance than others fibers. Currently, natural fibers composite have two issues that to be addressed: the zinc compatibility and water absorption. The chemical treatment has also been promoted to improve the bonding characteristic and reduce the water absorption characteristic. However, this method still questionable. The true mechanism of glyco hair addition and engineer has attempted for fabricate this scented fibers adhesive, giving even highly addition strength. However, high adhesive strength is not only one criteria in making performance of this fiber. Then we need more also consider about the repeatability, directionality, reliability, and efficiency. Natural fibers derived from plants mainly consist of cellulose, hemicellulose, lignin, pectin, and other waxy substance, as we can see from the tables. Cellulose is a highly crystalline structure which contains as much as 80% of crystalline regions. Hemicellulose is made up of highly branched polysaccharides attached to the cellulose after the removal of pectin. Lignin is amorphous, stiffen the cell wall, and act as a protective barrier for the cellulose. The basic chemical structure of cellulose in a plant-based fibers are similar, but they have different degrees of polymerizations, whereas the cell geometry of each type of cellulose varies with the fibers. As example, pulp contains cellulose ranging between 60 to 75% of cellulose. Different fibers, different growth conditions, and different testing method used by different study means that it's difficult to present one table that lists all the fiber properties. However, common chemicals compositions of some lignocellulose fibers of some are listed in this table. Cellulose was first isolated from plant matters by Miss Perrin in 1859. He reported that cellulose has an identical structure as starch, but it exhibits different in structure and properties. The physical and chemical aspect of cellulose has been extensively studied. However, in wood structure, cellulose can be found in the cell wall of plants, and the orientation of cellulose normally in vascular bundles was considered as a framework in order to support any applied external force. Nowadays, its unique hierarchical structure no longer holds any secret. Utilization of cellulose in various applications require a proper investigation of its psychochemical characteristics in order to understand the chemical structure and physical behavior. For plants, the amount of cellulose and its extraction will vary depending on plants to plant, soil condition, environments, as well as lifestyle. Case-by-case study of cellulose extraction and its derivatives should be individually employed. Today, cellulose is commonly known as a polysaccharide with the common formula C6H10O5N and consisting of a linear chain of several hundred to over thousand of linked glucose units. The worldwide production of this biopolymer is estimated to be between 11 ton each year, and only about 6 times 10 power of 9 ton is processed by paper, textile material, and chemical industry. The degree of polymerization is approximately 10,000 for cellulose chain in nature and 15,000 for native cellulose content. We can see from the figures exhibits the chemical structure of cellulose. Type of nanocellulose. The term nanocellulose broadly refers to three distinct types of fibrils. One, cellulose nanofibers, which is C and F. Second, cellulose nanocrystals, C and C, and bacterial nanocellulose, or B and C, each having different dimensions and properties. In general, tempo-mediated oxidation developed by Osogai in 2011 is the most popular method to extract cellulose nanofiber, or C and F. from wood pulp. It produces nanofiber of between 5 to 16 nanometer in diameter and several microns in length. In addition, CNC is obtained by strong acid hydrolysis of cellulose pulp, dissolving the amorphous part of the fiber while keeping their crystalline domains intact. The extracted CNC are of 5 nanometer in diameter with length between 20 to 100 nanometer. Depending on the type of cellulose, source, and treatment process, the size of both CNC and CNF is varies. Lastly, BNC can be synthesized from different bacterial strains, including Gluconase, Tobacter, Xylinus, Acrobacterium, Rhizobium, and so on. Xylinus is the most common bacteria used for BNC production. BNC has average diameter of 100 nanometer and length of micrometers. It is secreted by bacteria as an extracellular product of fermentation while cultivating in a glucose culture media. Fabricating of puff nanocellulose using tempo-oxidized cellulose. The natrium hydroxyl plus urea created tempo-oxidized cellulose with a high degree of oxidation was prepared to produce puff nanocellulose using a two-step process as shown in figure. During the first step, the hydrogen bonds in the cellulose 1 are destroyed by combination of sodium hydroxide and urea solution, and the individual cellulose chain with decreased crystallinity are obtained. During the second step, highly selective oxidation of the hydroxymethyl group of individual cellulose to the carboxyl groups was achieved by using tempo, sodium bromide, and sodium hypochlorite. Sodium hypochlorite acts as a primary oxidant with the catalyst amount of sodium bromide and tempo. Then, as can be seen from this figure, the synthesis of puff fiber using tempo oxidation process to obtain nanocellulose is shown. Okay, now we look for the infrared analysis of oxidized cellulose using FTIR spectrum. This FTIR spectrum of cellulose before and after oxidation was shown in this figure. In the spectrum of unoxidized cellulose, which is figure A and B, it can show that the value of wave numbers equal to 1064 is assigned to zero stretching cellulose backbone and is not influenced by the tempo-mediated oxidation. This peak can be treated as the internal standard, while from figure C and D, a new band appears 70-30 centimeter minus 1, which corresponds to a C double prime O stretching vibration of carboxyl acidic form. It can be suggested that the hydroxymethyl groups of the D-glucose unit were converted into carboxyl group successfully. The intensity of urea treated tempo oxidation solution cellulose is stronger than the direct tempo oxidized cellulose. It is indicating a high amount of carboxyl group and high degree of oxidation. Then we synthesize the puff nanocellulose into the hydrogel. Actually, the puff nanocellular hydrogel can be synthesized through the one-step method, which was dissolved directly in sodium hydroxide urate urea aqueous solution using cross-linker. In general, cellulose fiber can be covalently cross-linked with chemical cross-linker like citric acid, metal ion, or succinic anhydride. In this study, we use epihydrochlorine hydrin ECH as a cross-linker. Okay, then we try to run the experiment using SRD pattern for gelatin the hydrogel using the heating process at 50 degrees for 20 hours, which is, we call it an gel H2. And also we conduct the freezing process for minus 20 degrees Celsius for 20 hours, which is gel F2 as shown in figure. Gel H2 and gel F2 display similar diffraction peak at 2 theta equal to 20 degrees and 2 degrees, which correspond to the 110 and 200 plate of cellulose to cream form. Apparently, the crystalline of gel F2 is higher than the gel H2. This may be due to greater aggregation of cellulose chain when the hydrogel are prepared from relative higher temperature. However, transparent of the hydrogel from the freezing method was lower than from the heating process. The freezing method may lead to the increase of crystalline region within the three-dimensional work structure, which resulted in a loss of light transparency. As a conclusion, puff fiber can be synthesized to be a nanocellulose. Hydrogel also can be obtained from puff nanocellulose by using ECH as cross-linker and manipulated by heating and freezing methods. And finally, hydrogel has a potential to be used as a drug delivery, CD cell culture, and tissue engineering. That's it from me. Thank you very much.
Okay, thank you very much. We just watched the pre-record video presentation from Dr. Hassan. So please give him a round of applause by clicking the clapping hand on your screen. Thank you, sir, for sharing knowledge with us. So for all participants who have any questions, so please keep your question. We will have Q&A session after second invited speaker presentation. For all of you, also may write down your question at the chat room during the next presentation. Okay, ladies and gentlemen, we will have a second invited speaker coming from Dr. Munir El-Akabi. Let me read the short bio of the Associate Professor Dr. Munir El-Akabi. He was born in Morocco. He is now an associate professor at Material Science, Energy, and Nanoengineering at Muhammad VI Polytechnic University, or UM6P, and he had PhD in Polymer Science and Engineering at Maskik Foundation and Faculty of Science, Muhammad V Agde University, Rabat, Morocco. He have current research about the cellulose derivative and also lignin-based polymer, and also exploring the application of cellulose derivative such as cellulose fiber, cellulose nanocrystal, cellulose nanofiber, and also biopolymer in novel advantage. So, ladies and gentlemen, to keep the schedule on time, we will have presentation from Dr. Munir El-Akabi with the title Advanced Cellulose-Based Nanomaterial from Preparation to Multifunction Application. So we would invite Dr. Munir, please. Time and scale are yours. You can start, Dr. Munir, please.
Hello, you hear me?
Yes, sure, clearly.
I'm going to share my presentation. I don't have access to share my presentation.
Okay, so sir, you can share by yourself the presentation file.
Yeah.
Okay.
Do you see my presentation?
Yes, sir. Time is yours, please.
Yeah, okay. Good morning. Bonjour, salam alaikum. First of all, thank you to invite me to this international conference, the second international conference of Lignocellulose. So I am Professor Moel Al-Shabi from Mohammed VI Polytechnic University in Morocco. Especially I'm working in materials science and nanoengineering departments. Today I will give a short presentation on advanced cellulose-based nanomaterials from preparation to multifunctional applications. Mohammed VI Polytechnic University is a young university in Morocco. She's constructed in 2010 recently, and it is the leader of research in Morocco. I put here the website of the university to check and to see all the information related to our university UM6P. I'm going to present our research activities. We will give highlights on our research activities at the team. We are working on the extraction of cellulose from lignocellulosic biomass, and we use them for developing some advanced applications for industrial use. So let me start by some information related to plastics, because the final use of cellulose is in the plastic industry. Cellulose is biopolymers. And we know all that biopolymers are plastics. Hello. And use is
okay, sir, you can continue, please.
Okay, sorry. Plastic is used in almost every industry in society in our life, and the industrial use of plastics is almost in textile, and the use amount is used in packaging, and also in building, construction, consumer and industrial products, and others such as electronic and other applications. And the plastic is used in every day in our life. But the problem here by using the plastics is the recyclability. Almost more than 75% is not recycled today in the environment, and which is a very big problem for the environments, and the problem here is to replace the plastic or synthetic plastics by biodegradable or biopolymers to solve this problem for the environment. So, as I said, synthetic polymers are now biodegradable materials made from oil, resource, and after their use, they are not biodegradable and we have witnessed and the takeover of our world by plastic because they are not biodegradable and they generate very huge problem for the environment. Our strategy is to replace this. They cannot move. Our strategy is to replace the plastic-based products by biopolymers, which are biodegradables and biosourced materials. They are abundant, renewable, and eco-friendly and non-toxic biopolymers. And this is not to replace all the synthetic polymers, but parts of the use of plastic can be replaced by biopolymers. Here, just to move this. Insights on cellulose was presented before by Dr. Hassan. We know all that the main cellulose sources is wood and also some identified new lignocellulosic biomass. The cellulosic fibers is generally composed of lignin, hemicellulose, and cellulose fibers, which is here the lignin is played the role of adhesive to make the compact structure of cellulose. So from these raw fibers, we can extract many forms of cellulose. We can extract pure cellulose microfibers, but at the same time, we can also extract cellulose nanofibers, also nanofibers or nanofibrils. Maybe they are the same depending on the size and the properties. And finally, we can extract cellulose nanocrystals. We can extract all these forms from raw lignocellulosic biomass, depending by use, by controlling the processes and also the experimental conditions. Even if in laboratory scale or industrial scale, we have all these cellulose forms extracted from lignocellulosic biomass. They are now extracted at laboratory scale and also in industrial scale for many industrial applications, and also advanced researches are now in progress to use all these cellulose forms for testing them, for developing new applications, for especially for sustainable applications. The applications or the applications of cellulose or nanocellulose are today they are used in many sectors. They are used to develop composite materials. They are used also in biomedicine applications, for developing paper and pulp, for packaging, technical packaging and food packaging, and also for hydrogels applications, as presented before by Dr. Hassan. Also, they are used for construction, for water treatments, electronic and sensors, also for paint and coating. We note here that the conventional source of cellulose is the wood, and also the cotton, and the market of cellulose is always in progress. Here at Morocco, we have the surface of forest does not exist 13% of the territory. We have a small surface regarding the country. We have only 30% of the forest. So our research is focused to identify some the cellulose resources that are available in Morocco. We have in Morocco, we have by our team, we have identified some resources like alpha plants. We have more than 4 million hectares of alpha fibers, and also we have sugarcane bagasse. Also, we have hemp stalks, joncs plants, dome and palm fibers. Also, we have post-harvest eggplants, pavron and tomatoes plant residues. And also we have vein shoots and algal biomass. So we have identified all these resources in Morocco for extracting cellulose and nanocellulose and other molecules like lignin. So from this lignocellulosic biomass, we developed many final products like cellulose microfibers, cellulose nanocrystals, cellulose nanofibers, cellulose nanospheres, and also lignin biomolecules. And also we developed some final products like cellulose-based biofilms for food packaging, and also hydrogels and monoliths, especially used for wastewater treatments. I will give more details on this. So the outline of this presentation, I will give some processes applied to extract micro and nanocellulose by controlling the surface functionality and dimensions. And also I will give highlights on the applications of cellulose and microcellulose or functionalized cellulose and nanocellulose for coating of fertilizers to develop slow release fertilizers, and also the use of nanocellulose for food packaging, food packaging materials, and also the use of nanocellulose or functionalized nanocellulose for flame retardancy materials. So the applied process for producing the cellulose pulp before extracting nanocellulose. So we start from our starting materials is lignocellulosic materials, as I presented before. Of course, the first preparation is based on the grinding and pre-treatment of raw materials to prepare nano. To prepare ground fibers. Then we apply alkali treatments by using alkaline solution to prepare alkali fibers, and then we applied a bleaching process by using citric acid for preparing pure cellulose microfibers. Then we, from cellulose microfiber, we apply chemical processes such as acid hydrolysis to extract many forms of cellulose nanocrystals. We use hydrochloric acid to prepare hydroxylated cellulose, and we use a mixture of citric acid and hydrochloric acid to produce carboxylated cellulose. And also we use phosphoric and sulfuric acids to produce phosphorylated and sulfonated cellulose nanocrystals. So by applying different kind or different types of cellulose, we extract many forms of cellulose with different surface functionality and also with different dimensions, especially mostly we found from all identified cellulose resources in Morocco, we found that the form of nanocrystals are needle-like shape, with the diameter from 4 to 10 nanometers in general, and length of 200 to 500 nanometers, and with crystallinity more than 85%. Also, at the same time, we work on the extraction of cellulose nanofibrils, which is the second category of nanocellulose. We applied tempo-mediated oxidation process, starting from cellulose microfiber or previously extracted cellulose microfibers. We applied tempo oxidation process to modify the surface of cellulose microfibers and to facilitate the mechanical disintegration of oxidized cellulose into nano cellulose, and we apply then the mechanical disintegration or high pressure homogenization to produce cellulose nanofibers with oxidized surface. These cellulose nanofibrils are with diameter of from 1 to 5 nanometers in diameter, and with crystallinity more than 80%, and carboxylate charge contents of about 1,000 millimole CO2 per kilograms. Also, the second process applied by our team is the electrosonic processes. We start from carboxylated cellulose microfibers to produce cellulose nanospheres. Here we applied chemical treatments, as I presented before, And then we applied epoxidation of cellulose microfibers, and by using a simple process of ultrasonication, we obtain cellulose nanospheres with a diameter of 30 nanometers, sphere-like shape, and with a very important crystallinity, which is more than 70%, and with carboxylated charge, negative charge surface, with carboxylated charge content of almost of about 1,000 millimole per kilogram. And this is a very important process to produce cellulose nanoparticles with sphere-like shape. Also, we produce regenerated cellulose spheres with cellulose chip 2 by using phosphoric acid as solving solution. So we start from cellulose microfibers. We dissolve of cellulose microfibers in phosphoric acid at 5 degrees C for more than 10 hours in the agitation. And then we produce a clear solution of dissolved cellulose. And by adding water, we regenerate the cellulose spheres with stable colloidal suspension, and the analysis give us that are the produced or the originated cellulose is in form of cellulose spheres with the diameter around of 500 nanometers and with crystallinity more than 70%, and also with phosphorylated surface. Also, we developed another processes to produce sulfated micro and nanocellulose fibers by using sulfation process of cellulose, by using sulfation process of cellulose in sulfuric acid urea solution. We start from cellulose microfibers. We impregnate our cellulose in sulfuric acid urea solution, and we apply cooling at 150 degrees C for 1/2 hour to produce solid form of sulfated cellulose microfibers. And by washing and centrifugation, we produced sulfated microcellulose with the fibrous structure, and then we applied the mechanical disintegration process, which is called as high pressure homogenization, to produce sulfated nanocellulose with nanometric scale and with sulfur charge contents more than 2,000 millimole per kilogram and with crystallinity more than 70%. Also, we are developing, we developed another process to produce phosphorylated micro and nanocellulose fiber by using phosphorylation process by using fertilizer grade phosphates. We start from our microfibers. We applied treatments in DAP fertilizers solution in presence of urea, because the phosphorylation in literature is used by using commercial DAP products, but in our team, we used DAP fertilizer grade because we have OCP company in Morocco that produces DAP fertilizers, and we optimized the experimental conditions, especially the ratio between cellulose, urea, and DAP to produce phosphorylated micro cellulose fiber with fibers functionalized with the phosphate group with phosphate charge of more than 70 millimole per gram and with crystallinity more than 60%. We applied mechanical process or mechanical disintegration process to produce phosphorylated nano cellulose with crystallinity more than 60% and phosphate charge continents above 7,000 millimole per kilogram. This is all processes applied by our team and developed by our team here at UM6P to produce many forms of cellulosic fibers in micro and in nano and in nano size scale. Then I will move now to the application of nanocellulose for some applications. We use cellulose nanomaterials as coating formulations to develop slow release NPK fertilizers. So from lignocellulosic biomass, we extracted many forms I presented before. We extracted many forms of nanocellulose to coat fertilizers or NPK fertilizers for sustainable agricultures. We know that the problems related to fertilizers are they are highly soluble. The nutrients losses during the applications of fertilizers in soil. In general, the plants only assimilate a few percentage of nitrogen, phosphorus, and potassium, and the risk is lost in the environment, which generates a problem for economic and environment problem. So the application of the coating process of fertilizers is to the solution here is to reduce the loss of nutrients, slow release rates. So in this case, the plants are able to take up most of the fertilizers. So we produce by coating fertilizers with high efficiency, reduced labor capital, less frequency applications is required, and decrease risk of environments. So I will give some examples to use cellulose as coating materials. In general, this technology exists for many years ago, but we have today commercial polymer coated fertilizers, but all these commercial products are based on thermoplastic polymers, which are not biodegradable, and each year we have more than 15 kilograms per hectare can be accumulated in the soil each year, which generates a problem for the environment. So in our case, we apply cellulose as biodegradable materials to produce coated fertilizers. So we applied many kinds of cellulose and nanocellulose, and we use fertilizers produced by the Moroccan company OCP, and we applied two processes: fluidized bed process and coating pan process to coat the fertilizers by cellulose-based formulations. The objective here is to to develop slow release fertilizers. We look here that without coating, our fertilizers is soluble quickly, and when we apply the coating, the phosphate is released slowly. The same for nitrogen release. Here we introduced cellulose nanocrystals with PVA matrix to develop bio-nanocomposites based on nanocellulose up to 15%, and the thickness of the coating is about 10 micro. The objective here is to our control, our fertilizers is a fast release into 2.5 days, but when we apply the coating, the release of phosphate or nitrogen is up to 30 days, which is called slow release fertilizers. Also here, the coating by using biopolymers and by using nanocellulose have another advantage, which is water retention property. So the coated fertilizers could effectively store rain or irrigation water and improve the use of water resources for the soil and the plants. And also we evaluate the agronomic evaluation of the use of cellulose nanomaterials based formulation for fertilizer coating. We
Excuse me, Dr. Munir. I'm sorry
Yeah.
for interrupting you. Your time is 3 minutes left.
3 minutes.
Thank you. Sorry.
Okay, I will go quickly. So we evaluate economically the efficiency of our products in terms of leaf and the roots application, and also we are working on the use of nanocellulose nanomaterials for developing bio-nanocomposite for food packaging applications. So by using all cellulosic fibers or cellulosic types extracted from lignocellulose to develop products for food packaging applications. Especially we use cellulose nanocrystals and cellulose nanofibers combined with biopolymers to develop films for food packaging applications. Here, one example of using cellulose nanocrystals extracted from garlic spin and incorporated in chitosan biopolymer to produce bio-nanocomposites with the high mechanical properties, and also they have good antimicrobial properties because cellulose nanocrystals and some extracts from lignocellulosic biomass can give us this this very important antimicrobial properties for food packaging applications. And also we use cellulose nanocrystals or functionalized nanocellulose for bio-nanocomposite development for flame retardancy applications. Here, example of using phosphorylated nanocellulose incorporated in PVA matrix to develop biofilms, which are strong and flame retardant films. Here we some results on heat release rates, which is decreased by using cellulose nanofibers. The mechanical properties also are very important and strong. And also we developed, we are working on development of foams produced by functionalized cellulose nanofibers as flame retardancy materials. Thank you to my team here at UM6P, which is called Polycom team, and thank you for your attention.
Okay, thank you very much, Dr. Munir for really great talk about the nanocellulose. So, ladies and gentlemen, we come to the Q&A session. We will have 5 until 10 minutes. So for all of you who have any question, you can click a raise hand icon on your screen. Do we have any question? Okay, Miss Kanti, you can give a question directly to Mr. Dr. Munir.
Okay. My name is Kanti. Hello, Dr. Munir and Dr. Zaki. I have a question for both of you. To Mr. Zaki first. Based on your presentation, PALF was potentially used in many ways of fiber using, especially for hydrogel. The question is there any weakness or disadvantages or advantages found in using PALF as hydrogel rather than other raw material? And for Dr. Munir, what is the future challenge and opportunities of this cellulose-based nanomaterial? Thank you.
Okay, thank you, Ms. Kanti, for your question. Unfortunately, I think Dr. Zaki was left the seminar. So maybe please, Dr. Munir, for the answering Ms. Kanti question.
Yeah, thank you for this question. For the future challenge for the use of cellulose. Cellulose is a good product. Cellulose and its different derivatives and its different forms is very, very good materials. Especially, we can functionalize it. And we can control all the forms, all the structures, and we can develop many structures for with good properties for industrial use. But the future challenge is how we can produce all these cellulosic forms and all these cellulosic derivatives at industrial scale for industrial applications. This is the big challenge in the future because when we apply mechanical processes or chemical processes to produce all these cellulosic forms and derivatives, the price of these processes is expensive. The challenge is to find other ways and efficiency ways to produce cellulose derivatives and forms at industrial scale, but with shaper process. Currently, all the research is oriented, is focused to use less expensive processes to produce cellulosic forms and derivatives at lab scale, but we have to produce all these materials or biomaterials at industrial scale with shaper processes. Thank you. Hello?
Yes, thank you, Dr. Munir.
Thank you, Dr. Munir, for answering my question.
Okay, thank you, Mr. Munir, and also Ms. Kanti. So for the Dr. Hassan, I can phone you again back to the seminar. Could you please answer the question from Ms. Kanti? Okay. So while waiting for a response from Dr. Hassan, do we have another question, maybe for all participants? Okay. Regarding to the research about nanocellulose, actually in Research Center for Biomass and Bioproducts, Dr. Munir, we also have many researchers focused on the same field like you, nanocellulose for any application such as food packaging and other. So maybe I have one question. Dr. Munir, according to your slides, so is there any success story that, how do I say, the nanocellulose-based product, it was already converted to the industrial application that have a good response to the, how do I say, people in Morocco? Hello?
Yeah, till now, we are identifying that we are working on the identification of cellulose resources in Morocco. We have 10 years of experience in this field. We developed many products, but at lab scale. Now we are working out two startups. The startups, the first one is the production of paper and technical packaging from cellulose resources available in Morocco. This is the first one. The second one, we are working on the commercialization of nanocellulose extracted from our resources. But not for industrial use, but for research use, for lab use, for research use. So in general, we are working on to these startups, paper for industrial use and nanocellulose for research use. Our products will be commercialized maybe next years.
Okay, thank you, Dr. Munir. As you know, that nanocellulose is very, how do I say, interesting study, but we still struggle in the, how do I say, the low demand and also how to apply in the industrial scale. So thank you very much, Dr. Munir, for really good talk. So due to the limited time, so we are sorry that we have to close this session. Thank you very much for Dr. Muhammad Zaki Hassan and also Dr. Munir Al-Kabi for really great talk. We hope that maybe in the future we can do any collaboration research about the nanocellulose. So thank you very much for all participants for really great talk, for really great interactive. So before we close the session, we would like to remind you that we will have after lunch break, we will have come back again. Please keep in the room 1. We will start again at 12:30 Indonesian local time. So thank you very much. So have nice lunch time.
Thank you.
Thank you, Dr. Munir.
Thank you, Dr. Munir.
Recording stopped. Recording in progress.
Assalamualaikum warahmatullahi wabarakatuh. Tes.
Waalaikumsalam.
Can we start for this session, Bu Kanti?
Yes.
Okay.
Let's start.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon for everyone. My name is Sukma. I am a moderator for afternoon session for the second ICONLIG 2022. So in this afternoon session, we have the 15 presenter or speaker about the lignocellulose. So now we will divide it become 3 sections. So in the section 1, we will have the 6 presenter, and then continue for the other speaker, right? So we will the presentation in this session we will start for the speaker 6 speaker for the presents, and then after all of the presenter talk in this section 1. We will have the discussion, yeah, with question and answer in the last 6 presenter in the section 1. So each presenter have the 10 minutes for the talks, and then we have the about 20 please wait, sorry, about 15 minutes for the discussion. So let's start for the section 1. So can we show the short bio for the 6 presenter in the section 1, Bu Kanti? So in the section 1, the presenter are the Pak Ahmad Sauki, Bu Novitri Hastuti, Bu Sri Feni, Pak Muhammad Rashidur Ridho, Bu Depi Nurani, dan and the Pak Ali Rahmat. So in the first presenter is Pak Ahmad Sauki. So I will show the short bio of Pak Ahmad Sauki. So here we can see this wait. Pak Ahmad Sauki is senior lecturer at Department of Biology in University Islam Malang, and he graduated from Environmental Resource Management and Development in postgraduate of Brawijaya University. The current research are mainly on looking for the organic plant material for raw material of bioethanol. And the second presenter is Dr. Novitri Hastuti. She is the researcher at the Research Center for Biomass and Bioproducts BRIN, Bogor, and also he graduated from the Kyushu University, Japan, for the doctoral degree, and now he earned the postdoc through the Fulbright Visiting Scholar 2022 in this years at the University of Wisconsin Madison, and he will talk today about the microfibrillated cellulose from agar waste in alginate-based hydrogel for papain enzyme immobilization. And the third presenter is Ibu Sri Penny Wijayanti. She is a researcher at the Research Center for Sustainable Production System and Life Cycle Assessment. She graduated from Chemical Engineering at Gajah Mada University, and current research is mainly on sustainable bio-industrial and green chemical technology. And the fourth presenter is Muhammad Rashidur Ridho. He is a master student of Forest Product Science and Technology at IPB University, and also he is now a research assistant in bioproduct-based polyphenol research group from Research Center of Biomass and Bioproduct, joined in 2020. And he will talk today about the lignin and particle board. In the fifth presenter is Devi Nurani. She is a student at Brawijaya University and majoring in the agro-industrial technology. Now also she is a research assistant at the Laboratory of Biomass and Bioproducts, and he will talk about the almost similar with the previous presenters. And the sixth presenter in the section 1 is Ali Rahmat, PhD. He is a junior researcher at Research Center for Limnology and Water Resources. He graduated from Gifu University for the doctoral degree, and current research is mainly on soil and water conservation. So for the first time, I will invite Pak Dr. Ahmad Sauki to present your topic. Please, Welcome. Bu Kanti, how about the for each presenter? Whether we will play the video or directly or live present?
Of Lignocellulose. I am Ahmad Shauki, corresponding author. This paper has title Rapid Test Bioethanol Raw Material, the indigestion data method modification to fiber cellulose. The paper, many authors. This is my name and Majida Ramadan, Akinta S. Anggraini, Hera Yuwana, and Fatmawati from Department of Biology, Islamic University of Malang, in Malang, Indonesia. Our paper has attention to lignocellulose and as intrinsic organic is the plant's residual for one of among the environmental conservation way like composting. Applying saccharification of the lignocellulose aimed at raw material of bioethanol production as a key in the all production generation. The current study and ready to propose biosaccharification with microorganisms, that mean from fungi, Aspergillus niger, Hansenula, Saturnus, Candida utilis, and Trichoderma viridae. Generally, it is known cellulose and hemicellulose is polymer and suspected source of glucose. This for fermentation. However, it has various methods to eliminate lignin because the material has content lignin variation. Or to open the lignin, this term is delignification. The gap with previous research, it is known yet about around residual analysis for presuming the quantity glucose source in plant residual. Therefore, it needs knowledge method immediately to suspected polymer that is a source of glucose. How to find out the cellulose content, which can be categorized as a simple and quick method, all type of plants. This research has aimed to compile a rapid analysis method for cellulose from resources of all environmental sectors uses the glucose hydrolysate parameter. These materials and the method, the focus for experiment with design of one short case study. Independent variables in this research: first, chemical agent, varies concentration of sulfuric acid, a long time of soaking, using sulfuric acid 72%, initial hydrolysis concentration of sulfuric acid 0.5 molar and 72%. Dependent variable, mole of glucose using the formula with molar evaluation. Mole of glucose equals 6.944 times 10 power minus 4 times Brix, percent Brix, and times the volume of the hydrolysate and times the residual mineral divided by the weight of this residual. This diagram and result in discussion. The implementation was performed the windy method to say Papetandra, and the yield is not different, this analysis, and this side from Putra. It's not different, I think. And then chemical agent using the hydrochloric acid and then sulfuric acid. This for test of hemicellulose, 10 persons, and then 8 persons from Abdel Fattah and Abdel Nabi side in the journal. The fiber that contain cellulose, hemicellulose, this result of analysis person fiber quantity, and it's around the quantity from this side, Ringgita Liman Irwanto. And comparing glucose yield, initial of the concentration of sulfuric acid and the implementation for 2 hours is not different. This the implementation of 2 hours and this standard. And the initial using the concentration of 72%, it's highest error. This is the process to find the times of soaking, 2 hours. What the argumentation using 2 hours soaking for hydrolyze the residual? This similar pattern, 4 hours and 2 hours. The hexose from hemicellulose is lower than cellulose. This pattern is similar. And then this for implementation soaking in sulfuric acid, ananas leaf and banana stem. The implementation for ananas leaf is consistent with previous to find how many hours soaking in sulfuric acid. And banana stem, this hexose from hemicellulose is higher than cellulose because this different the content of lignin, I think. Summary this presentation. Compiling method. First, using Wien-de-Jessen method to open the cell wall component, especially the lignin, by sodium hydroxides. And then to hydrolyze the source of glucose in cellulose fiber using 2 hours with sulfuric acid in the initial hydrolyzing 0.5 molar and then 72%. This is the end of the presentation. Thank you. And wassalamualaikum warahmatullahi wabarakatuh.
This next video, Bu Kanti.
It is a pleasure for me to be here in the second International Conference of Lignocellulose, ICONLIG 2022. And I am Novitri Hasuti, a researcher from Research Center for Biomass and Bioproducts, National Research and Innovation Agency. Today I would like to present my work entitled Microfibrillated Cellulose from Agar Waste in Alternate-Bed Hydrogels for Papain Enzyme Immobilization. So this work, I did this research with my team, with Ibu Luciasi, Ibu Safrina, Pak Wahyu, Ibu Dian, Ibu Maya, and also Miss Aisyah Taskiatunisa. The outline of my presentation will be introduction as the first part, and the second part is primary goals, followed by materials and method, and including the formula to make a hydrogel, and the fourth part is results and discussion, and ended by conclusions. Enzyme is important materials in biotechnology. However, the enzyme performance is affected, or this enzyme is very susceptible to the environmental conditions such as temperature, as well as pH, and etc. And the using of enzyme in the biotechnology or biological process is important. Therefore, several studies to improve the enzyme performance, as well as to explore the material that can bring enzyme in a safe template or safe carrier, have been explored and investigated. Cellulose as fibrous materials are the most promising solid supports for protein and enzyme immobilization due to their height-specific surfaces as well as porous structures. And cellulose as fibrous materials also providing the quantity and accessibility of the active sites that's required to drive the reaction rate of enzyme itself to get the more effective or more economical conversion in a biological or biotechnology process. The objective study, beside we did a characterization of the agar waste itself and about the microfibrillated cellulose that we get from the chemical process. So the primary goals of the study is to provide a new perspective utilizing cellulose from industrial waste to be proceed or to be managed more technologically and utilized in more advanced application in this study, particularly in enzyme immobilization. Materials and method of this study, we start from pulping process using sodium hydroxide 10% for 3 hours at 100 degrees Celsius. So we get the delignified cellulose, and we did the bleaching process using hydrogen peroxide and washed by water. So we will get more clean and white cellulose. And then the cellulose, the neutralized pulp or the neutralized cellulose, we put into the bigger glass to do the hydrolysis process using sulfuric acid 40% at 45 degrees Celsius for plus minus about 1 hour and a half or up to 2 hours. And then to make the hydrogel, we put the several concentration from the microfibrillated cellulose that we get from the hydrolysis process, reacted with the alginate, and we put the calcium chloride as cross linkers so that we can get the hydrogels containing the papain enzyme. So this is the formula to make the hydrogels. We have 4 treatments here, denoted as N0, N1, N2, and N3. N0 hydrogels containing alginate and enzyme, as well as calcium chloride, but there is no microfibrillated cellulose inside. And for the N1, N2, N3 containing microfibrillated cellulose about 0.25% and 0.50% and 1% respectively. So after getting white bleach and wash pulp, we got the white pulp and we did the observation under a microscope. So this is the same images of the raw pulp, I mean the bleached pulp or the cellulose after the pulping process. And after sonication, hydrolysis and sonication, so the sulfuric acid process can defibrillate the agar fibers to the smaller and smoother fibers with the smaller scale bars here, we can see here. And this picture can confirm that the hydrolysis of 40% of sulfuric acid at 45 degrees Celsius is able to defibrillate cellulose of maize agar. Under particle size analyzer, we can see the distribution of the size that we get from the hydrolysis process. So the results of the particle size analyzer saw that the fiber diameter of 300 up to 900 nanometer dominates in our suspension, and this result also confirms that acid hydrolysis is able to defibrillate the cellulose of agar waste. We did the analysis of swelling ratio for each treatment, and then from this table we know that a sample containing about 0.25% of microfibrillated cellulose, they had a highest swelling ratio close to 92%. And to measure the protein release from the hydrogels, at first we make a standard curve of bovine serum albumin or BSA, and then we can plot the data so we can get the intercept and the slope values to calculate the protein release from the hydrogels. And this is the results. We did the measurement for 2 periods, which are 1 hour and 24 hours. So from these results, we know that the treatment containing a hydrogel containing 0.25% of microfibrillated cellulose tend to the lowest release of enzyme in 1 hour period, and after 24 hours, so the protein release is relatively lower compared to the hydrogel containing the microfibrillated cellulose higher than 0.25%. So from this study, we conclude that the utilization of microfibrillated cellulose from waste agar showed that the concentration of 0.25% in the alternate matrix is had the highest swelling ratio, around 92%, with a better immobilization compared to the other alginate hydrogels containing microfibrillated cellulose higher than 0.25%. And however, we need to explore about the optimum concentration lower than 0.25% to determine the optimum concentration of microfibrillated cellulose as a hydrogel scaffold for enzyme immobilization. That's all my presentation, and thank you for your kind attention.
Okay, next video, Bu Ganti.
Assalamualaikum warahmatullahi wabarakatuh. My name is Silviani Wijayanti. I'm from Research Center for Sustainable Production System and Life Cycle Assessment. Today, I would like to present my research about evaluation of an alginate chitosan microcrystalline cellulose sulfate microencapsulation system for efficient fermentation of lignocellulosic hydrolysate. As we know, lignocellulosic material is a cheap renewable resource that is available in large amounts and contains cellulose, semicellulose, and lignin. Lignocellulose also can convert the fuel ethanol through hydrolysis and fermentation process. Using sulfuric acid at high temperatures and pressure is one of the fast and cheap methods for acquiring sugars from lignocellulose, but produce more inhibitors. The toxicity of inhibitory compounds inhibit the activity of yeast from converting sugars to ethanol. One of the immobilizing methods that are successful to reduce the inhibitory effect is encapsulation. In encapsulation, we use alginate, chitosan, and cellulose sulfate. Sodium alginate, water-soluble salt, is gelled when it comes in contact with bivalent cation, such as calcium ions, but the reaction is a reversible process. To increase the chemical stability of the calcium alginate is by coat it with cationic polymers such as chitosan. The polyelectrolyte complex between alginate and chitosan is considered irreversible and formed only through weak ionic associations. The reason was that although large amounts of amina are protonated, there are chloride ions that may be counterbalancing the charge. So only a small portion of this protonated amino group are available to interact with the carboxylate groups of alginates. Because this reason, the research intends to modify the association by introducing either covalent bonds or additional conjugating species. In this study, the conjugating species chosen are sodium microcrystalline cellulose sulfate. Next is experimental design. We started research with the preparation of sodium microcrystalline cellulose, then continued to encapsulation and fermentation. For the preparation of sodium microcrystalline cellulose, like on the screen, we use a sulfation agent, chlorosulfonic acid, and dimethylformamide, added to the activated microcrystalline cellulose. The quantitative characterization was analyzed by NMR and infrared spectroscopy. The value of the degree of substitution was calculated by conductometry. Next, the capsule were prepared by the platforming one-step method. This is the composition of the capsule membranes. Before the anaerobic cultivation, cell-seeded capsules were aerobically cultured in the defined growth media with a glucose to propagate a high biomass concentration inside the capsules. Anaerobic cultivation was then carried out in conical flask placed in a cycle bath. Hydrolyzed media was adjusted to pH 5 by adding sodium hydroxide. Then the appropriate amounts of all the mineral salts and the trace elements were added to make the same composition as in the synthetic growth media, except for the carbon source already present in the hydrolyzed. Samples were withdrawn from the flask during the experiments, centrifuged and stored at minus 20, then HPLC was used to quantify the metabolites. Sulfation of microcrystalline cellulose was performed to prepare the polymer that was used as conjugating species in the encapsulation of yeast. The presence of sulfate groups in sodium microcrystalline cellulose was confirmed by the infrared spectra. The analysis of sodium microcrystalline cellulose by NMR was also done to analyze the position and distribution of the sulfate group. This is the estimated structure molecule of sodium microcrystalline cellulose from the experiment. Okay, the degree of substitution of the sodium microcrystalline cellulose represents the number of sulfate groups in the anhydroglucose units. The conductometric titration has been chosen to measure the degree of substitution since the method was cheap, easy, and given a reliable value. The degree of substitution of sodium microcrystalline cellulose was only 0.1, which means there was only one sulfate group in 10 monomers of the anhydroglucose unit. The different physical properties of sodium microcrystalline cellulose and microcrystalline cellulose sulfate were the solvent used to dissolve both compounds. Sodium MCS could easily dissolve in water, where MCC could not dissolve in water. Sodium alginate is a viscous solution that has a lot of negative charge. When the sodium alginate is added to a calcium chloride solution, the calcium ion displaces the sodium ions and form a gel. Cytosine has been chosen as adding a layer for alginate capsule. Cytosine has amino groups which will be positive when the cytosine dissolved in acidic acetate buffer solution. The alginate cytosine microcellulose sulfate polyelectrode complex was made by combining alginate and MCS polymer in different concentration. When using 0.06 MCS, the spherical capsule was successfully formed, but when the concentration was increased to 0.50%, the spherical capsule were difficult to form. Since the degree of substitution was low, there was not enough negative charge binding with calcium ions. When we assume that the presence of a sulfate group will increase the mechanical and chemical stability of capsule. However, the electrostatic interaction to form the alginate cytosine MCS layer was weak. Finally, only alginate cytosine MCS formed using 0.06 MCS, mixing with 0.54 sodium alginate was used for further fermentation study. Acid hydrolysate contain many inhibitor compounds that will damage cell viability for fermenting sugars to ethanol. No growth was observed when the free cells were exposed to 50% hydrolysate media, and the glucose consumption and the ethanol production rate were deficient. The glucose consumption, ethanol production, and ethanol yield of encapsulated cells were higher than those of free cell cultivation. Reducing the concentration of calcium chloride from 300 to 25 millimolar increased the rate of glucose consumption, ethanol production, and ethanol yield. Capsule stability increased with decreasing concentration of calcium chloride in the cytosine. A high calcium chloride concentration decreased the membrane hydrophilicity and increased the membrane porosity. Decreasing the hydrophilicity of the capsule membranes can reduce the amount of glucose entering and increase the number of toxicity compounds. So ethanol product is low. Overall, adding sodium microcrystalline cellulose to alginate cytosine layer caused no significant increase in the glucose consumption rate. The ethanol production rate on the ethanol yield in the hydrolysate media. Okay, and then the conclusion is, the conclusion are that capsulated cells have been investigated by alginate cytosine and alginate cytosine MCS capsule membranes to prevent yeast from inhibitory compounds. The capsule's composition has influenced the cell ability to produce ethanol. High calcium chloride concentration of cytosine solution harms the capsule membrane due to the high porous membrane, allowing the inhibitors to pass easily. Investigating the effect of using NIMCS with higher degree of substitution may be worthwhile, since this may improve the membrane quality to produce a high amount of ethanol. Thank you very much for your attention.
Okay, next presenter, Bu Kanti. Please play the next video. Bu Kan. The voice, Bu Kanti.
I will present the result of our research entitled Isolation and Characterization of Lignin from Black Liquor of arecanut leaf seed with hydrochloric acid and phosphoric acid.
The background of this research is the area of arecanut plantation in Indonesia, approximately 147,000 hectares. From the arecanut plantation, its product waste that has not been utilized by farmers, one of which is the areca leaf seed, as much as 43,000 tons per year, usually just thrown away and burned. The lignin content in biomass varies depending on the type of plant. For arecanut, the lignin content is 19.59%. One way to extract lignin from biomass is the lignification process. Samples were pulped with active alkaline or AA and sulfidity. The delignification process product black liquor and pulp. Then the lignin is isolated from black liquor by adding acid, which can be organic acid or inorganic acid, and then obtain lignin powder. To the best of our knowledge, there is not a report about lignin isolation from craft black liquor of areca leaf seed. This study aims to examine the properties of lignin from black liquor product by pulping areca leaf seed using the craft method at different active alkaline or AA loading 17 and 19% with 20% of sulfidity loading. Meanwhile, inorganic acid, that is hydrochloric acid and phosphoric acid variations were used in lignin precipitation from black liquor. The material we use in the research are areca leaf seed from PT Grini Alam Indonesia, sodium hydroxide and sodium sulfate for the craft pulping process. Hydrochloric acid and phosphoric acid was used for lignin isolation. The lignification process of areca leaf seed. Areca leaf seed chip of 500 grams were boiled for 3 hours to remove extractive substance. A craft pulping process we carried out with active alkaline or AA loading conditions of 17 and 19% and sulfidity loading of 20%. The pulping was set a temperature of 170 degrees Celsius for 2.5 hours. After the process was complete, the pulping was separated from black liquor by a screen sheet and then stored in closed bottle. The whole process of lignin isolation can be seen on the slide. First, the black liquor product from the pulping process was isolated for lignin using different acids, namely hydrochloric acid and phosphoric acid. Next, lignin deposit was separated from the supernatant by centrifugation. The resulting lignin was adhered using an oven at 60 degrees Celsius for 24 hours. then crashed using a motor, and then the referred alkyl lignin was characterized among them lignin yield, chemical component, FTR, PWGCMS, and TGA. The result of the analysis of the chemical component of lignin can be seen in the table 1. Hydrochloric acid yield a higher lignin yield than that of phosphoric acid. Ibrahim et al. 2004 state that using different acid in lignin isolation may affect the yield. For lignin purity, phosphoric acid product a higher purity of 54.07% for AA 17% and 43.79% for AA 19%, while isolation with hydrochloric acid obtained with lignin purity of 29.29% at AA 17% and 31.82% for 19%. Using the different acid in the acidification process with a lower pH of the liquid can make a higher silica content and obtain a lower quality of lignin. The difference between acid and pH product different IL decrease in pH to 4 result in a higher IL than pH 2. A lower pH can lead further degradation product than different from lignin in the isolation process. As can also affect the purity of lignin. The higher the as content cause, the lower purity of lignin. Isolate lignin, hydrochloric acid, and phosphoric acid product higher as content than of the lignin reference. Lubis et al. 2014 state that the high as content in lignin is due to the presence of impurity caused by the delignification method and the type of acid used in the lignin isolation process that can increase impurities. The primary functional groups in lignin are carbonyl, carboxyl acid, hydroxyl, and methoxyl, but the functional group of lignin varies depending on the lignin source. The functional group of the lignin that was isolated using different acid and AA at a pulping process are quite similar to that of the reference lignin. It can be seen in the figure 1. Several functional groups detected in isolate lignin are described in table 2. As we can see in the table 3, the different types of acid do not affect the Sg ratio, but the difference is the concentration of the alkali active. Sg ratio of lignin with AA treatment was 19%, higher than lignin in AA treatment, 17% for hydrochloric acid and phosphoric acid. Lignin with treatment was 19% for phosphoric acid, with the higher proportion of syringil in its effect the high thermal stability of lignin. In figure 2, we can see the graph thermal analysis of lignin alkyl using the TGA. There is 3 stages of lignin decomposition. This result suggests the isolated lignin had a higher thermal stability due to higher charge remaining at 750 degrees Celsius. The aromatic ring on lignin degrades slowly. The more stable thermal characteristics are, the higher the mass remaining after the most recent combustion. Handika et al. 2021. The conclusion from the result of this study: first, the highest lignin yield was obtained in lignin isolation by hydrochloric acid compared to lignin phosphoric acid. Second, lignin priority of the total IL and NaSl was highest in phosphoric acid lignin compared to lignin hydrochloric acid. Third, the ash content of lignin was isolated with hydrochloric acid and phosphoric acid that is higher than that of reference lignin. Fourth, isolate lignin from different acid and different AA has a relative similar functional group corresponding to unique lignin peaks such as syringil and guaiacyl rings. And fifth, thermal analysis on arecalypsid lignin makes a higher mass residue than lignin reference, except for phosphoric acid lignin with AA 17%. Thank you for the opportunity.
Next video, Bu Kanti.
Assalamualaikum warahmatullahi wabarakatuh. I'm Adia Nurani as a representation. Today, I want to present our research about the properties of cellulose nanofibril produced from different sources, pineapple leaf, and oil palm fronds. Introduction. Recently, industry development focused on eco-friendly biomass derived from nature. Biomass can be derived from agricultural waste. Indonesia has a large agricultural land which contributes to increase in agricultural waste. Pineapple leaf and oil palm fronds are two agricultural wastes that have a large amount in Indonesia. 1 hectare of farmland can produce 3 tons pineapple leaf during harvest time. Meanwhile, oil palm fronds which can reach to 3.108 tons. Pineapple leaf and oil palm frond have a high cellulose content. The cellulose content of oil palm frond is 34.89% and that of pineapple leaf is 66.20%. So cellulose has the potential to be used. Cellulose can be transformed into a nanocellulose, one of the classification of nanofibril or CNF. CNF of fiber with length in micrometer and diameter in nanometer. CNF can be used in many applications such as pharmaceutical, drug delivery, packaging, and tissue engineering. Because the CNF structure consists of an amorph and crystallinity part which have biocompatibility, high exposure ratio, high surface area, tensile strength, and coefficient thermal expansion. To obtain cellulose nanofibril with good properties can be infused by the isolation method and characterization of raw material. In general, mechanical treatment is used to reduce the size of the fiber. Previous studies have been conducted to isolate CNF using mechanical treatment. The resulting CNF has a high crystallinity index, which affects nanocellulose thermal stability. Mechanical treatment can also reduce fiber size from macro to nanoscale. As a result, the goal of this research was to create and characterize CNF from various sources, such as pineapple leaf and oil palm fronds. The mechanical grinding process has been used to obtain CNF from bleached pineapple leaf and oil palm fronds pulp. The properties of CNF were analyzed using field emission scanning electron microscope for morphology, zeta potential to determine surface charge, and the rheology to determine viscosity. And also the raw material was determined by the chemical compound. Material and method. Chemical composition. The raw material was also determined by the chemical composition, including moisture content, ash content, and then alpha cellulose content, total lignin content, and hollow cellulose content. Isolation cellulose nanofiber will start with soak 2 grams of pulp pineapple leaf and oil palm fronds in 2000 ml distilled water at a consistency of 1%. And then the slurry of the fiber will thrown in supermass collider Masuko Sangyu grinding with gaps of -10 micrometer and -30 micrometer for 3 pass each gaps. And then the result is CNF from pineapple leaf and as a CNFP and CNF from oil palm frond as a CNFO. And there are 3 characterizations using field emission scanning electron microscope for morphology, and then HORIBA with laser Doppler electrophoresis technique for zeta potential and barometer for viscosity. And then result and discussion, determination of lignocellulose composition show that pineapple leaf pulp have a 64.68% cellulose and oil palm frond pulp have 59.114% cellulose. It indicates that pineapple leaf pulp have a higher cellulose content than oil palm frond pulp. And then pineapple leaf pulp have 2.478% of lignin content, and oil palm frond pulp have a 4.774% lignin content. It indicates that the pineapple leaf has have a less lignin content than the oil palm fronds. The hemicellulose content show that pineapple leaf have a higher than oil palm fronds, and the bleached oil palm fronds pulp has a higher moisture content and ash content than the bleached pineapple leaf pulp. And the cellulose nanofiber CNFP and CNFO show that have an appearance of shell-like because the fiber can reach nanometer scale fiber size. Whereas CNFP has a white color and CNFO has a light brown color suspension. The difference in the color of suspension CNFO from bleached pulp is due to oil palm frond still have a high lignin content. The suspension of CNFP and CNFO didn't settle after a few days. It's indicating that the suspension has a good dispersion in water. And then the morphology. The morphological structure of CNFP and CNFO were observed using field emission scanning electron microscopy with magnification of 120,000. The morphology structure of CNFP and CNFO show that the fiber have an irregular structure and elongated round. The diameter of CNF can determine using IMAGE software, and the result CNFP has average diameter 53.7 nanometer and CNFO have average diameter 56.72 nanometer. The main characteristic of cellulose nanofibril is it has a long fibril with length in micron and average diameter less than 10 nanometer. And the next is zeta potential. CNFP have a zeta potential value minus 52.6 and the CNFO have a zeta potential value minus 41.6 millivolt. The suspension of CNFP and CNFO has a good stability because the result of the zeta potential value CNFP and CNFO greater than plus minus 30 millivolt indicates that the suspension have a good colloid stability due to sufficient repulsion. And the negative value in the zeta potential determine the presence of electrostatic repulsion to indicate the stability of the colloid suspension. The effect of mechanical treatment can cause many hydroxyl group in cellulose being exposed, therefore negative charge on the surface increase. And the rheology analysis show that average viscosity of CNFP and CNFO were 131.68 millipascal and 114.11 millipascal respectively. The viscosity will be decreased when the shear rate increases. At the same shear rate, viscosity of CNFP is higher than the CNFO. It may be caused by the smaller fiber size of CNFP. The smaller size of the fiber increase the surface area, therefore it makes the solution cohesive and viscous due to interaction between the particles. Several things that affect viscosity CNF are particle size, concentration treatment, and pH in the cellulose nanofiber. And the conclusion, cellulose nanofiber was obtained by mechanical treatment with Masuko Sangyo ultrafine grinder. The appearance of CNFP and CNFO like gel, and both of them have a good stability. The morphology CNFP and CNFO has an irregular structure. The zeta potential of CNFP and CNFO has a good colloid stability. And the last, the viscosity of CNFP higher than the CNFO. That's enough for me. Thank you for attention.
Okay, next video, Bu Rizka, please play the. Bu Rizka, could you please play the next video?
My name is Ali Rahmat. I am from Research Center for Limnology and Water Research. Today, I want to present my research with the title Chemical Properties of the Coconut Lubber Sawdust Biochar using X-ray fluorescence under low temperature pyrolysis. Indonesia is an agriculture country, which is in the agriculture activity will produce a lot of the residues. For example, branch, stem, small tree, wood chips, sawdust powder, which is all of them is the major source of the biomass. Wood sawdust is the biomass industrial waste that is created by the furniture industry and other wood processing business. Wood is chopping, sizing, resound, smooth, which is producing the sawdust. From the 100 kg of wood, will produce at least 12 until 25 kg of sawdust. In this slide, we can see the picture of the coconut timber of lubber, which will be processing for the furniture and will produce the sawdust, where abundance of the sawdust to wind which frequently blow and suspend the wood dust in the atmosphere and will impact on the air quality. The air pollution will make a problem on the human respiratory. So we need a solution to minimize this problem and this risk. One of the strategies to control this problem is converting the sawdust to be biochar, where biochar have a lot of the advantage for the soil improvement. What is biochar? Biochar is charcoal resulting from the heating of the biomass in oxygen-limited environment. In this picture, we can see the charcoal from the various types of the source of biochar. Why biochar? Biochar is 2,000-year-old practice that convert agricultural waste to soil enhancer, which can hold the carbon, increase the food security, and discourage deforestation. Biochar can be important tools to increase the food security, cropland diversity in the area with severely depleted soil, scarce organic source, and inadequate water and chemical fertilizer supplies. Also, biochar is the powerful, simple tools to combat climate change. So the benefit of the biochar is are enhance the productivity of farmlands, achieve environmental friendly agriculture, organic agriculture, sustainable carbon sequestration, rural promotion, and environmental friendly education. So, biochar, the quality of the biochar depends on the raw material and the process of the pyrolysis. So the purpose of this research is to investigate the chemical properties of the coconut rubber sawdust derived biochar under different temperature of pyrolysis. The methods. So the raw material will be furnace in the furnace for 4 hours. After that, we can see the next is the carb already be biochar, and we ground and sieving. After that, we will analyze using XRF. Result. From the analysis of the XRF, we can see the highest content of the coconut wood sawdust biochar if the Fa or iron, which is around 49 until 50%. After that, silica, and then aluminum, and then calcium, kalium, and phosphate. So, if we see the content of the biochar, is questionable because this is questionable. It is potential or not if we apply the biochar in the soil, because the concentration of the Fa is quite high, where Fa is micronutrient, but if we give a lot of Fa, will be toxic to the plant or the crops and maybe for the environment. So the composition of the Fa and Se also Al also is the toxic element if the concentration too high, even though the calcium, kalium, and phosphate is the macronutrient which is needed for the growth of the plant. I think from this research we can use the biochar as another material, maybe for the concrete or other material which is need a lot of the Fa and Se for the main material. So the conclusion, the highest content in the term of the macronutrient of biochar is calcium, around 7 until 12%, then potassium around 4 until 5%, phosphorus around 1 until 2%. However, the concentration of Fa is high, around 46 until 50. Also, silica, also L is 8 until 10%. It can be toxic if applied to the soil. On the other hand, coconut rubber sawdust can be source of the silica due to the silica content being quite high, like 19 until 20%. Maybe will be used as the nanosilica or other material. And thank you for your attention. I am Isali Rahmat from the Research Center of the Limnology, graduate from the Gifu University and currently research in soil and water conservation. And now I research utilize the agriculture waste to improve the soil quality and also to improve the water condition. Thank you very much.
Okay, participant, we still have around 15 minutes or maybe less than 15 minutes, yeah. for the discussion. So please, if you have any question for 6 presenter in the section 1, please raise hand. Is there any question? Okay. Okay, Bu Kanti, please unmute your voice.
Okay, thank you for the opportunity, Mr. Sukma, and thank you for all the presenter, such enjoyable video presentation. I want to ask to 3 presenter first, to Mr. Rido. The question is there an explanation about how different acid used for isolation affect the yield of lignin, and based on the result of your research, which acid treatment you recommend to isolate lignin best? And to Mr. Ali Rahmat, based on your research result, the concentration of Fa or iron is high, so and it is toxic for soil. So what kind of application of biocharcoal will you suggest, and why? The last one is for Mrs. Sripeni. If we compare the regular fermentation of ethanol, which one of the most valuable in terms of stability, yield, and cost effectiveness? Thank you.
Okay, we're waiting for other questions. So please answer the question from Bu Kanti. For Mr. Rido, the first, please answer the question. Mr. Rido, could you please open your video?
Okay. Thank you for the question. Lignin as a hydrochloric acid product, the highest yield, but the purity of lignin is low. It can also be seen that the ash from the lignin isolation product value about 14%. Will isolation as phosphoric acid product small yield about 10%, but high lignin purity of approximately 13 until 14%. I would recommend it as phosphoric acid because it is an hydrochloric organic acid and product high yield.
Okay, thank you, Mr. Rashid. How was Bu Kanti? Is enough or still?
The first question is not answered clearly. I think my question, the first question is there an explanation about how different acid used for the isolation affect the yield? So the yield from your result is really have a really big differences. So is there an explanation about how come different acid make such a big difference in yield?
So, Mr. Rido, could you please answer the reason about your result? Or still have search the reason? Okay, it's okay, Bu Kanti, ya. Maybe in the next time you can discuss more detail. Okay. The second question delivered for the Mr. Ali Rahman. Please, Pak Ali, could you please answer the question from the?
Thank you. Thank you, Dr. Sukma, and thank you, Dr. Kanti.
It's because this is due to the reactivity of different acid protonation.
Oh. Okay, Mr. Rido, all right, thank you for your answer. Bukanti, okay. Sorry, Pali, interrupt. Please, Pali,
Never mind.
answer.
Okay. Thank you very much for the question. Thank you. Currently, my research team is trying to search the good biochart based on the nutrient contents. And unfortunately for biomass from the coconut tree luber, they have high Fa, but we also have from the jati wood as a source. They have the high calcium, like 50%, kalium 15%, phosphate until 10%, and no let's say toxic element or very low toxic element. In others practice, usually we introduce the farmer at least try using the rice husk or sekam, because they also, even though they have the high silica contents, but sometimes silica is needed by the paddy crops. So actually, in this and from our research, if we want to get the biochar with the high potassium or kalium content, the good source is from the seed waste. And if we want to get the good or high content of the calcium, from the source is good from the wood waste or sawdust from the woods. I think that's all. Thank you.
Mr. Ali, I mean, if you not recommend the biochar of coconut lumber sawdust for soil because it's toxicity, so what kind of application of this coconut lumber sawdust biochar will you suggest? I mean, for another application besides for the soil.
Okay. Maybe I cannot answer clearly. Maybe they can be used as the absorbents. Maybe I don't know. Maybe they have the good like porosity. Maybe because I'm not doing the advanced research, because we now only check the chemical properties like that. This is not sustainable or not suitable for the biochar. Maybe can be substitution as sorts like the carbon absorbent, maybe activated carbon, and I don't know. This maybe can be like for the additional material for concrete. Maybe I'm not sure because that's not my not my major, not my expertise. But that's maybe that can be used for others material. But for now, based on the chemical properties, not recommended to apply to the soil. I think that's all. Thank you.
Okay, thank you,
Okay,
Mr. Ali.
clear, right? Bukanti. Okay. Next, Bu Penny, could you please answer the question from Bukanti? Bu Penny still drive. Bu Penny? Still mute. Please unmute your microphone.
Yeah. Thank you, Ms. Kanti, for your question. I am not clear about your question and what you mean about regular fermentation. Is it in the defined ground media or what?
Your presentation is about the wait a minute.
Setelah 300 meter, belok kiri.
So your presentation is about the alginate microcrystalline for the fermentation.
Yes.
And due to the expensive process, So if we compare to the regular fermentation of the ethanol, which one is the most valuable in terms of the stability, yield, and the cost effectiveness, maybe?
It's mean without encapsulation.
Yes, without the
without
encapsulation.
the encapsulation. Yes, okay. If we compare it without the encapsulation, of course, maybe I'm not I calculate the techno economy maybe I'm not calculate for the economy. Is it valuable or not? But I think maybe because we need to process encapsulation first and then make formulation after we have capsule. I think it will be not valuable if we compare with the regular fermentation, maybe.
And what about the stability and the yield of the ethanol itself, Mrs. Penny?
The stability, okay, the stability if we use the hydrolyzed lignocellulose, the stability, of course, the stability of fermentation, I think it will be good if we if we protect the yeast from the toxic media. I think the stability of yeast, if we use to for fermentation, it will be stable. Then if we not to capsulate.
Setelah 200 meter, tetap di kanan di pertigaan. Ikuti rambu kapal
Of this answer, what you expect.
Okay. Okay, thank you, Bu Penny.
Yes.
I think it's clear ya, Bu Kanti ya. Maybe we can discuss more after this session. Okay. Because of the time, I think the discussion in the first section we will the end, and we will continue for the next section, the section second section. So in the second section, the speaker is are the Pak Modi Lempang, Bu Efrida Basri, Pak Muhammad Yusron, Bu Dwi Ajias, and Bu Esther, also Pak Bu Masyuni. So could you please, Bu Rizka, show the short bio of their presenter? So Pak Modi Lempang is a researcher at Research Center for Biomass and Bioproduct. He graduated for the master degree at IPB University, and the current research is about the timber forest and non-timber forest product technology. And the second presenter is who is, please, next for Bu Rizka, Pak Saifuddin. He is a researcher at Research Center for Biomass and Bioproduct and graduated from the Pakuan University for the master degree. And the focus research is about the bioprospection of natural materials. And the third presenter is Ms. Fahria Puspitasari. She is from the Research Center for Biomass and Bioproduct and graduated from the Department of Chemical Engineering, the Diponegoro University. Now, her focus research is the valorization of lignin, active packaging, antioxidant, and UV barrier materials. And the next presenter is the Dwi Ajias Pramasari. She is the junior researcher of polysaccharide engineering research group at Research Center for Biomass and Bioproducts. She graduated for the master degree and bachelor degree from the Agro-Industrial Technology IPB University. And now her focus, next. It's okay, never mind. The next presenter is Esther Rima Suryani Toga Torov. She is from the research assistant at Research Center for Biomass and Bioproduct BRIN. She graduated, still study. Graduated from the Bioprocess Engineering from Faculty of Agriculture Technology, Brawijaya University, for the bachelor degree. The topic of this session for her is on biomass-derived carbon for functional materials. And the next presenters in this section is Professor Dr. Masyuni. She is from Department of Chemistry, Faculty of Mathematics and Natural Science, Halu Oleo University, and she is graduated from the Gajah Mada University, right, for the doctoral degree. And she have the many patent, a national patent for her research result in the research. And the focus of the research is about nanocomposite membrane. Biosensor design and others, maybe. Maybe we will announce detail after her presentation. So we can start for the first presentation in this section 2. So please start. Pak Modi Lempang.
Thank you for the opportunity to present my research paper entitled Properties and Potential Use for Paper Pulp of 4 less known hardwood originated from natural tropical rainforest in South Sulawesi province of Indonesia. Let me start with introduction. In 2020, Indonesia's log production has only reached 59.27 million meter cubic, of which of 47.99 million meter cubic was for chip and pulp production. From the pulp, paper and paperboard are made. Until now, the main raw material for paper pulp is wood. Indonesia's pulp production from wood raw materials in 2020 was 8.59 million tons. Although wood resources have decreased significantly, globally paper demand continues to increase. Paper and paperboard production from pulp globally in 2015 was 406.3 million tons. Wood paper needs are estimated to increase by more than 100 million tons per year. The paper pulp industry generally use only certain species of hardwood like mangium, eucalyptus, or sengon, which are monoculture and are harvested by clear cutting, so the utilization causes forest degradation and impact on surrounding environment. Some species of wood that potentially yet as raw material for patient natural tropics. The properties of the pulp and paper produced from these species can be better than those produced from mangium and eucalyptus. Utilization of as much as possible of wood, including less known wood species, are widely open because the availability of good quality wood for industry raw material is increasingly limited. This presented paper is the result of research fiber quality and chemical components of 4 less known hardwoods and their potential use for paper pulp. Methods, material. 4 species of less known hardwoods, namely Cempeda, Jabon Merah, Saling. All species originated from natural tropical rainforest in South Sulawesi province of Indonesia. Figure 1 indicates that tree leaf, bark, and and bark of cempeda, saling-saling, jabon merah, and simpur. Sample preparation and measurement. Trees of 36 to 50 centimeter in diameter for each species were used as a test sample. Wood sample in this form of 5 centimeter thickness taken from the base, middle, and top of tree stem. In figure 2, indicate the wood sample in this form of cempeda A, saling-saling B, jabon merah C, and simpur D. Specific gravity. Measurement of specific gravity using a test sample size of 4 by 4 by 2 centimeter. The specific gravity be determined by the weight of the open dry wood and the volume of air dry wood, where the wood volume be measured by emission method. The specific gravity were classified based on Iowa criteria. Fiber dimensions. The dimension of fiber are measured through maceration preparation, which was made using the plant clean procedure. Dimension of fiber measured include fiber length, fiber diameter, and lumen diameter. The quality of woods were assessed based on Indonesia hardwood fiber quality classification for pulp and paper raw materials compiled by the Agriculture Department of Indonesia Republic. Chemical analysis. From wooden discs were taken several snippets. The snippets then grounded and shaped it to obtain wood powders. The wood powders which passed the 80 mesh shape and were retained 60 mesh were used as sample for chemical analysis. The chemical analysis uses the TAPI agreement method. The chemical analysis were cellulose, pentose, lignin, extractive, and ash. The chemical contents of wood were assessed based on Indonesian hardwood chemical classification compiled by the Agriculture Department of Indonesia Republic. Result. Specific gravity. Specific gravity of 4 less known hardwoods ranged from 0.40 to 0.69. All species classified as wood with moderate in specific gravity. Woods that have a specific gravity of 0.55 to 0.65 is prepared as raw material for paper pulp. Fiber dimension and quality. The wood fiber of cempeda, jabon merah, and saling-saling are classified as long fiber, whereas wood fiber of simpur is classified as very long fiber. Requirement of wood as paper pulp raw material are long fiber, fibrous. Fiber length is a determinant factor of paper strength, like tear index, tensile index, and folding index. Fiber wall thickness of jabon merah and saling-saling classified as very thin, while fiber wall thickness of simpur classified as thin to thick. The fiber walls of thin to thick will be quite easy to flatten when mild, and very long fibers will produce strong waving power. Fiber quality of less known hardwoods. Total score of fiber length and fiber dimension derivation of 4 less known hardwoods ranged from 400 to 450. And the quality of fiber all classified in class 2. Chemical component. The most important chemical component of wood for paper pulp is cellulose. The cellulose content of 4 less known hardwood is medium to high. Potential use for paper pulp. Based on wood properties of 4 less known hardwoods, all species are potentially for paper pulp production. Although all of 4 species studied represent the potential wood resource for paper pulp production, but CP is highly recommended due to this species has very long fiber and higher specific gravity. Conclusion: from natural tropical rainforest in South Sulawesi province of Indonesia are not yet be used as raw material for paper pulp production. In condition to paper pulp raw material, the result of this study provides an understanding that based on wood-specific gravity, fiber quality, and cellulose content of cempedak, jabon merah, saling-saling, and simpur are represent a potential resource for applicating to paper pulp-based industries. Wood of simpur is highly recommended than 3 other less known hardwood species for paper pulp raw material due to very long fiber and high specific gravity. I end my presentation by saying thank you very much.
Thank you very much, moderator. I'd like to presentation about properties of oil palm wood Lignocellulose jack modified with bioimpregnant. Saifuddin represent 7 authors. Background: oil palm plantation could provide more than 30 million meter cubic of wood stem per year, but until now, the utilization of the wood is still limited to full pulp, especially the palm oil factory. The mechanical and physical properties low density, unstable dimension, compression strength, and perpendicular to grain. Grain properties tendency to external check and collapse. The aim of this study is to describe the physical, mechanical, and drying properties of oil palm wood, which impregnated with wood bark extract. Material and method: 25 and 32 years old oil palm tree and bioimpregnant wood sample were taken from the outer portion of lower stem. Bioimpregnant mixture of 30 years old bark extract, resorcinol resin, and formaldehyde. Mixture was added with 0.5 part of tapioca starch, flour, and natural hydroxide. The sample for treatment is shown below. A, high temperature drying properties, and B, determination of compression strength. Result and discussion. Oil palm wood, which were impregnated with bioimpregnant, significantly increases the density of compression strength, but parallel and perpendicular to grain in comparison with not impregnated wood. Perfect penetration is shown from the test sample impregnated with bark extract using vacuum pressure. Illustration from wood density, compression strength in the next slide. Impregnation oil palm wood, 25 years and 30 years old. Vacuum pressure treatment result was the highest density in the right histogram. Density of 30 years old specimen increased about 2 times. Compression strength parallel vacuum pressure treatment result was the highest. Vacuum pressure of 30 years old specimen increased more than 6 times than control of 25 years old. Compression strength perpendicular vacuum pressure treatment result was the highest, and compression strength of 30 years old specimen increased higher volume than other treatment. Visual color of wood with impregnation method. A. Control light gray. B. Hot impression gray, and vacuum pressure dark chocolate. C. Remarks: impregnant bark extract plus formaldehyde plus resolutional resin plus tapicostat fluor plus natrium hydroxide, and perfect penetration is shown in figure C. Drying properties. The drying properties of oil palm wood aged 25 and 32 years are not good. Oil palm wood impregnated with vacuum pressure process for 25 years age belong to rather low up to fair quality, while for 32 years age were categories is quite good quality. Drying properties from vacuum pressure treatment 30 years old had classification of drying defect 3 up to 4 quite good up to fair. Data shown in the next slide. Like this, treatment classification of drying defect, temperature minimal, maximal, and humidity maximal, minimal. Without impregnation control, classification of drying defect is 6 up to 7 are very low, and vacuum pressure of 25 years old is 5 up to 6 rather low, and vacuum pressure 20 years old 3 up to 4 quite good up to fair, and temperature minimal, humidity minimal, maximal is variation. Right. Conclusion: oil palm bark extract or bioimpregnant could increase the oil palm wood density, compression strength, parallel and perpendicular to grain. Treatment of bioimpregnant by using vacuum pressure resulted in high density and compression strength parallel as well as perpendicular to grain, then thus impregnated by using hot immersion. The increase of the compression strength of the impregnated wood by vacuum pressure was higher than those of impregnated by using hot immersion. Drying properties of vacuum pressure impregnated wood had rather low and quite good quality for consecutively 25 and 32 years on. The best drying schedule of the 25 years old impregnated with vacuum pressure 50 up to 80 temperature and 80 up to 12 humidity. Thank you.
Next, Bu Rizka.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. My name is Maria Puspitasari. I would like to present about our study entitled The Hydrophobicity Characteristic of Soda Pulp of Rice Straw Modified with Water-Soluble Chitosan. Our team are Muhammad Yusron, Azizatul Karimah, Muslimatul Rahmi, Mayang Aristika, Bagaskoro Twilight, Adi Ardana, myself, and also Widya Fatiliasari. We are from IBB University, Research Center for Biomass and Bioproducts BRIN, also from PT Solusi Biru Indonesia, and also from Research Collaboration Center for Marine Biomaterials. The background of this research: in 2020, 10 million hectares of rice is harvested, generating a huge amount of rice straw which is costly to replace and difficult to dispose of. Usually, to get rid of the leftover straw from the field, people just burn it. The utilization of straw must be optimized into value-added products to minimize the environmental impacts. Rice straw is a lignocellulose biomass that contains hemicellulose, lignin, and also cellulose. Hemicellulose could be used to produce sugar production, and lignin and other organic compounds can be utilized as a fertilizer to improve soil quality, while cellulose can be used as additional raw material in the pulp and paper industry. To optimize the utilization of hemicellulose, lignin, and cellulose, the rice straw must be pretreated. Pulping is a process of separating the fiber from materials containing lignocellulosic biomaterial. In some products, such as biomedical devices, the hydrophobicity of paper must be increased. The pulp must be modified to increase its hydrophobicity and strength. In this research, we try to use water-soluble chitosan, which is not only non-toxic and biocompatible with the human physiological system, but also can be improved the paper strength, hydrophobicity, and also optical properties. The aim of this research are to prepare and evaluate the hydrophobic rice straw pulp using the atmospheric hot soda pulping or EHSP process and soda pulping or SP process, and then modified with water-soluble chitosan treatment. For the materials and method, the rice straw was sun-dried for a day and cut into 2 to 5 cm. The pulp process was carried out by using 200 grams rice straw chips and adding the chemical liquor. We use sodium hydroxide 19.5% with a biomass to liquor ratio of 1 to 20. There are two variations of pulping process: soda pulping process and also atmospheric hot soda pulping process. The SP process was carried out for 1.5 hours at 170 degrees Celsius and 8 atm, while the EHSP process was carried out for 1.5 hours at 100 degrees Celsius and 1 atm. After the pulping process, the samples was filtered using a filter cloth and washed with water. The samples are stored in the Ziploc bag at 4 degrees Celsius. After that, we analyze the glycine lignin and also the carbohydrate content. After pulping process, To modify the pulp, we prepared by adding 10 milliliters of water-soluble chitosan solution with different concentration, 1% and 2% to a 3% of pulp suspension. The mixture was stirred for 10 minutes at 350 rpm and at room temperature. The wet modified pulp was dried in an oven at 60 degrees Celsius for 24 hours for water absorption analysis, and the remaining pulp was also dried by freeze drying at minus 50 degrees Celsius for 22 hours for functional group analysis. And for the hand sheet paper production, with a grammage of 80 gram per meter square, was made by preparing a modified pulp suspension was formed on a 10 and 10 centimeter mold. The formed hand sheet paper was dried in oven at 60 degrees Celsius for 24 hours. After we get the hand sheet paper, we analyze the water absorption capacity, contact angle, and also FTIR analysis. This is the result of our research. In this table, we can see the pulping properties of rice straw with atmospheric hot soda or AHS process and soda pulping or SP process. Based on the result, the yield of rice straw pulp with SP process was 22%, while the EHS process was 29%. The SP has high pressure and temperature, which can increase the reactivity of sodium hydroxide as a pulping chemical, so that the delignification process is more optimum. And along with the delignification process, the carbohydrate is also degraded. On the other hand, in EHS process, has low pressure and temperature, which causes lower reactivity of sodium hydroxide. And for the kappa number is can be indicate the remaining lignin content in the pulp, presenting the degree of delignification and also the level of maturity in the brightness level of the pulp. Pulp with a good degree of delignification will give a low value of kappa number in the testing. Based on the result, EHS pulping process has a higher kappa number than SP process. And for the class on lignin, which is indicating the remaining lignin content in the pulp. In this case, EHS process resulted in the higher residual lignin of 2% compared to the soda pulping of 1%. Lignin in the pulp generally has an undesired effect on the pulp quality because the fibers become stiff and difficult to fibrillate, causing low physical strength of the pulp. Also, the higher of the lignin content, the darker the pulp color. And for the delignification selectivity is used as a key parameter of the effectiveness of pulping process. Good delignification selectivity could produce a pulp yield that has high carbohydrate and lower lignin content. The delignification selectivity of SP process was 15, which is higher than EHS process. The difference in the delignification selectivity of the pulping process might be because of the chemical concentration, pulping temperature, and also pulping time. From this result, can be indicated that SP process has more effectively pulped the rice straw than EHS process. And we can see in this figure, this is the rice straw before and after pretreatment. This is rice straw before treatment, and this is the pulp of the rice straw after EHS process and also from soda pulping process. This is the result of the hydrophobicity testing we carried out to determine the water resistance of the pulp and hand sheet paper samples. In the addition of 2% chitosan to EHSP and SP pulp causes the decreased water absorption value compared to the unmodified pulp. The difference in the pulping process tends to affect the difference in the water absorption value. Pulp with EHSP has a lower water absorption value because of the higher content of lignin than SP pulp, because lignin has a hydrophobic and water resistant properties. And this figure, we observe the contact angle over time until 30 seconds. This is the figure from the unmodified pulp, and this is the figure from the pulp modified with 1% of chitosan, and this is pulp with 2% of chitosan. At the 30-second, the whole water drops were observed by the unmodified sample. It is a relatively rapid decrease in the contact angle, while some water were held on the modified sample. It shows that modifying pulp with addition of chitosan gave a significant influence of the water absorption. At the same duration of the observation, EHSP and SP paper sample with 2% of chitosan tends to have a very small change in the contact angle and the best surface resistant to water. The change in the contact angle from time to time tend to slower compared to sample with 1% of chitosan. Even though in the water absorption test, the SP sample with 1% of chitosan has the same value of the untreated sample. In the contact angle test, the SP with 1% of chitosan concentration has a value that tends to be lower than the control sample. This is because of the paper sample. Chitosan could have accumulated in the paper surface during production and could have filled the paper pores. Pulping differences tend to not affect the resulting in the contact angle value. And this is the K-value of the ratio of pulp sample. The K-value indicates the increase in the wettability of the samples. The low wettability values indicate the materials tend to have a hydrophobic properties. These results indicate that the higher the concentration of chitosan causes the decrease in the wettability on the paper surface. From the results can be concluded that rice straw pulp has been successfully prepared by atmospheric soda process and soda pulping process. The higher screen pulp yield comes from the HSP sample, about 29%. And SP pulp has a lower kappa number and higher delignification selectivity than EHSP pulp. Following the pulp modification, the amount of water-soluble chitosan in the modified pulp increases along with the increase in the hydrophobicity. EHSP and SP modified with a 2% water-soluble chitosan treatment have better hydrophobicity. That's all for my presentation. Thank you very much for the attention and have a nice day.
Okay, Bu Rizka. Next video, Bu, please.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for this opportunity to me to present our research in the
Wait a minute, Pak.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for this opportunity to me to present our research in the second International Conference of Lignocellulose or ICONLIG 2022. My research is about the sorghum Mycorrhiza carabica Y67 adaptation in sugarcane trace hemicellulosic hydrolysis for xylitol production. I am Dwi Ajes Pramasari from the Research Center from Biomass and Bioproduct as the presenter, and this is my co-author. Xylitol, or the sugar alcohol with the 5 carbon, it can be application in sweetener in food, odontological, pharmaceutical manufacture. And if we see the market share for the sweetener for the xylitol is around the 12.5%. And he has the unique characteristic, like this equivalent sweetness and lower calories, and the third is lower glycemic level. For now, the currently commercial xylitol production use the catalytic hydrogenation. We say that a nickel catalyzed method, but the energy and cost intensive process. And the other problem is the production is not environmentally friendly. So the researcher looking for the new technology. We say the alternative biotechnology technique by cellulose fermenting yeast. This is some of the yeast can be used to produce the xylitol. And in this study, we constantly use the Mycorrhiza carabica Y67 in ACC. From the previous research that mentioned that Mycorrhiza is higher yield in the xylitol production, and the second is good resistant to inhibitor. In this study, we use the sugarcane trace. This is one of the low-cost prospective lignocellulosic biomass you can to xylitol production, and she has the hemicellulose content is higher. From the data Hermiati et al. Is around the 35.50% of the hemicellulose. That it can be extraction throughout the hydrolysis for the sugarcane trace hemicellulose hydrolysate, and we can fermentate for the xylitol. By yeast, the high acidic environmental is the problem. So for the maintenance the perform of the yeast, we use the short adaptation in this study. We use 4 days with the performing a pH adjustment and use the Mycorrhiza carabica in ACC Y67. So that the aim for this study is to evaluate the effectiveness of short-term adaptation technique for obtaining the higher xylitol. We use the sugarcane trace, obtain the PT, PG, Rajawali 2 Subang. After that, we preparation the sugarcane trace for the raw material for the acetolysis. Use the microwave with the maleic acid. And after the sugarcane trace hemicellulose hydrolysate is ready, we adjust with the various pH like this and use that for the STHH fermentation media. After that, we adaptation of yeast cell or the yeast, the Mycorrhiza carabica. Before that, we enrichment with the isolate the starter, and we conducted the adaptation in 4 days. Each day, we use the new of the inoculum, and then after the 4 days in the various pH, we fermentation in the 3 days, and after the 3 days we analysis the xylitol concentration with the megazyme kit. This is the result and discussion for our study, and we can see the xylitol consumption profile was similar in all the evaluated adaptation condition, and with the control or not adapted. One of the factor for influence the successful bioconversion of xylitol to xylitol, one of them is extracellular pH has a significant influence on metabolic process as well as product formation. And we can see that during the 1 day fermentation, cellulose concentration did not increase significantly, but after that, after 2 days, it is decreased significantly. It's all of the adaptation yeast can be consumed around the 59 until the 63% of the initial cellulose. And we can see too that cellulose consumption profile in adapted strain is not different from the control or not adaptation, and it can be predicted adaptation can be more tolerance for compared than not adapted yeast. Concentration, we can see that the all adapted strain can capable to convert the cellulose to the xylitol at the low pH, and we can see the adapted yeast at the pH 2.5 is the produce the highest concentration. It's around the 0.48 gram per liter. However, it's lower from the control or not adapted growing at pH 5 is around the 1.48. But in previous research, not edited yeast in pH 3 and pH 4.8 using the corn crop hydrolysis did not produce the xylitol. And it is good for this condition. In the lower pH, it can be convert the xylose into xylitol. But not this is the production of xylitol can also be increased by using the proper amount volume of fermentation media. In this study, it still use 50 milliliter. So if we can decreasing the volume, and we can expect it the strength it can more adaptate. If we see the xylitol yield and the percentage of theoretical xylitol yield, we can see that the highest xylitol yield was adapted strength at pH 2.5, and then the lowest yield is from the adapted pH 3, and it is related with the percentage of theoretical xylitol yield. We can see that the highest is compared with the theoretical is the 5.53% of theoretical. And based on the data, xylitol productivity, generally it can be seen the short-term adaptation affect the bioconversion of cellulose into the xylitol. The highest xylitol productivity adapted yeast at the pH 2.5 is around the 0.01 gram per liter per hours, and this is the higher than the various adapted yeast in this research, and it is similar to the xylitol concentration and xylitol yield. In the previous, the productivity value was lower than the non-adapted yeast at pH 5 or the control. Therefore, we can conclude that our research is the short-term adaptation of yeast cell in the study can be enhanced xylitol production and can be effective method for improving yeast tolerance to the low pH of the acid hydrolysis as well as its fermentive performance. And the second, we can conclude that compared with the common range pH for yeast or the control in pH 5, short-term techniques still need some improvement to obtain the similar result. Thank you for the DIPA Life Sciences project for the funding. Thank you for the Integrated Laboratory of Bioproduct BRIN for the facility. And we also want to thank Aditya Nawati for preparation of sugarcane trace hemicellulosic hydrolysis. Thank you very much for your attention. Let me know if you want to know about our research and the question about this presentation. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, Bu Rizka, please continue for the next video, Bu.
Good day, ladies and gentlemen. Let's say thank you for your coming and the attention you gave for my presentation. Our study is about proximate and structural analysis of activated carbon with different structure from oil palm biomass. With me, Esther Rima Suryani Togatorop, and my friends Ria Yolanda Arundina, Prabu Satria Setaji, Sukma Surya Kusuma, and Resti Marlina.
Activated carbon is widely used as a filter, absorbent, electrode material, and etc. The conventional activated carbon is non-renewable and non-eco-friendly because the material is from coal. So we give solution biomass-derived activated carbon because it's abundant, eco-friendly, and is cheaper from the coal. Oil palm biomass is one of the largest agricultural waste in Asia that can be a promising commodity as activated carbon material. In addition, the conversion of biomass from agricultural waste into value-added product such as activated carbon is a creative solution for environmental problem solving. Morphology and structure of the raw material are important because it can affect the pores and the surface area of activated carbon. In this study, we used two kinds of raw material. One is oil palm antifreeze bunch or OPEFB, and another one is oil palm cell or OPS. Besides the differences of morphology and structure, we use two kinds of carbonization. Oil palm antifreeze bunch OPFB and OPS will hydrothermal carbonization, and the oil palm cell char will pyrolysis carbonization. Preparation and characterization. The first step is hydrothermal process. In this hydrothermal process, OPFB and OPS will wash and dry. After that, hydrothermal in 220 Celsius in hours with continuous agitation. After that, dry it in oven and crush until 200 mass, and then we get hydrochar OPFB and hydrochar OPS. The next step is activating process. In this process, hydrochar OPFB, hydrochar OPS, and OPS char will activating with KOH with ratio 1:1 for 2 hours at temperature, and then this mixture and solution of catalyst ferrum chloride were mixed for 5 hours at 60 Celsius in agitation. Then this mixture was dry overnight at 80 Celsius to prepare the impregnated sample. The impregnated sample was set in aluminum crucible, then pyrolyzed at 800 Celsius for 1 hour under nitrogen flow. And then the sample were neutralized using 1 molar hydrochloride solution overnight with continuous agitation, then washed with distilled water until the pH became neutral. The washed sample were dried at 105 Celsius for 24 hours to prepare the activated carbon. The activated carbon from OPFB as ACT, from OPS as ACC, and from OPS char as ACA. Then the activated carbon will analyze. From proximate analysis, we know the moisture content, ash content, volatile matter, fixed carbon, and absorption of methylene blue. The highest moisture content is in ACA, followed by ACC and ACT. The highest ash content was found in the ACT samples, followed by ACC and ACA respectively. ACT with the large volatile matter with value 12.36%, because ACT has a bigger hydrocarbon content than ACC. ACA volatile matter is lower than ACC. This is because activated carbon using hydrothermal has higher volatile matter, because pyrolysis allows dehydration and decarboxylation process to occur that evaporates oxygen in form of water, carbonyl, and carboxylic acid. The fixed carbon of activated carbon is formed in the range of 56 until 83%. In this study, the value of fixed carbon in ACA is the lowest. The best adsorption was in ACT, then ACC, and ACA. It was explained that the larger the surface area, the greater the absorption of methylene blue. The next is X-ray diffraction. From X-ray diffraction characterization, we'll get the XRD graphs that show all activated carbon samples have an amorphous carbon structure with two main peaks. The peaks is around 23 and around 43. From the graph, we can also see that there is no graphitization process. And from X-ray diffraction, we can see or we can count the crystallite size. SEC has 0.64 and SEC has 0.69, ACA has 0.7. From X-ray diffraction, we can conclude that pyrolysis method give highest crystallite size, and SEC from OPS has the bigger crystallite size than OPFB activated carbon. The next is absorption desorption nitrogen. The BAT graph shows that the surface area of ACT is larger than the SEC because the crystal structure of OPFB is more porous than OPS. Meanwhile, the surface area of ACA is lower than SEC because the pyrolysis process does not initiate the formation of new pores. The isotherm curves of 3 activated carbon is type 2 with hysteresis. This shows that activated carbon is dominated by micropores and has formed a mesopores. From Fasm analysis, can be seen that the pores formed in the ACA sample as a sample representing the pyrolysis carbonization have limited pores when and SEC. The same thing absorption. The using steam can increase the production of meso and micropores significantly compared to the pyrolysis that open pores and is followed by formation of narrow micropores. Fasm from ACT and SEC show an abstract surface and evenly distributed pores. This shows that the hydrothermal process affects the formation of more pores. From Fasm analysis, it can be concluded that the hydrothermal process has formed more significant pores compared to the pyrolysis method. From this study, we can conclude that we have success in synthesizing activated carbon from 3 raw materials, OPFB, OPS, OPS-char, with high fixed carbon value and good absorption ability. The best activated carbon was found in the ACT sample, with the highest absorption ability of fixed carbon, the lowest moisture and ash content. The initial morphology of the raw material can evoke the activated carbon form. It is proven by the result of absorption-desorption of ACT being higher than SEC. From XRD, it can be seen that the diffraction peaks formed in ACA are higher and sharper than SEC and ACT. ACT and SEC have a more uneven surface area and evenly distributed pores, while the SEC surface does not have many pores and the distribution is uneven. In this case, hydrothermal carbonization provides a larger surface area due to initiation of pore formation, while pyrolysis carbonization does not initiate, so that the surface area and absorption ability of activated carbon using the hydrothermal method is better. Activating carbon produced in this study can already be used as adsorption, filtration, and electrode material. Here are the ends of my presentation. I would say thank you for your attention. Good day.
Wabarakatuh. I would like to say thank you for the opportunity given to us at the second International Conference of Lignocellulose. The present our title result is GCMS-FCR analysis of coconut cell lignocellulose conversion using pyrolysis method and as a strong antifungal agent on cocoa seeds. The research by Masyuni M. Jahidin Laulika Didaye, Fitri Handeni Hamid, and Nur Arfayanti. We are from Department of Chemistry, Physics, and Biology, Halu Oleh University, Indonesia. Introduction. Cocoa or Theobroma cocoa is one of the most profitable commercial crops. Production of global cocoa is estimated at 4.59 million tons for 2017 and 2018. Cocoa plantations are the primary source of income for around 1 million farmers in Indonesia. More than half of these producers are small farmers in Sulawesi, one of the main islands in Indonesia. Production about 16 until 71% of nationality production. The The presence of pests and disease, especially cocoa fruit borer, has contributed to decline in cocoa yields. In 2009, the government of Indonesia established a revitalization program to increase cocoa productivity through pesticides and fertilizer to restore soil nutrients in cocoa farms and supply cocoa seeds. Farmers in overcoming fungal problems in cocoa seeds usually use synthetic pesticides. According to the World Health Organization, pesticide consumption is increasing worldwide. One of them is a pesticide that kill or inhibit the growth fungal causing disease in plants. Farmers generally widely use synthetic fungicides because they are easy to obtain and the use process is simple. The use of synthetic fungicide will have a negative impact because it can portion both the farmers as operator and the general public as consumer, change the ecological balance or agricultural ecosystem that can lead the pest resistance and cause the pollution to the environment. Since synthetic fungicide can have a negative impact, an upright alternative solution is the delivered toxic fungicide. Agents target microorganisms but not harmful to humans and animals around them. They do not pollute the environment because it is biodegradable and harmless to human health. Coconut shell is one of the organic waste from processed coconut fruit, which is lignocellulose biomass. Lignocellulose have three compounds are lignin, cellulose, and hemicellulose. The compound contained in lignocellulose can be decomposed by heat to form a condensable vapor that can continuously produced by oil at atmospheric pressure using pyrolysis. Pyrolysis technology is a thermal decomposition process occurred under oxygen-free conditions, which result in the chemical bond cleavage to form small molecule compounds. It is based on the thermal conversion of biomass in three chemical steps: char, by-oil, and gas. The by-oil as the primary product, such as alcohol, ketone, phenol, carboxylic acid. The chemical compound in by-oil from biomass can be used as antibacterial, antifungal, antioxidant, and anti-cancer. The coconut shell was cleaned from the fibers, cut into smaller slices of 3 cm and dried in the sun for 3 until 4 days. A dry coconut shell was put into a pyrolysis reactor. The coconut shell was pyrolyzed at temperature of 200 until 600 degrees Celsius for 3 until 3 hours. The resulting oil is filtered before providing the following analysis stage. Analysis of by-oil includes analysis of compound using GCMS and antifungal testing on cocoa seed using total plate count method. And then the characteristics of observation on cocoa seeds include water content and pH value. Result and discussion. The yield of coconut shell pyrolysis product at temperature of 200 until 600 degrees Celsius with a heat rate of 10 degrees Celsius per minute. Pyrolysis has three products, are by-oil, tar, and char. For the primary product produce by-oil yield of 35%. By-oil have pH value was 3 and density was 1.082 kg per cubic. The GCMS analysis of coconut shell by-oil. The result of the GCMS analysis showed that the most dominant compound was phenol and its derivatives. Phenol content of 23.18%. The FCER analysis of coconut shell biochar. On the wave number is shown C-C indicating the existence of small molecule from the secondary product of macromolecule. for the stretching fabrication of the CCC group, such as aliphatic and aromatic compounds. CH3 stretching fabrication will be related to alkane compounds, especially their asymmetric and symmetric. Stretching fabrication of CC, such as aromatic compounds, and then the CH group, such as aliphatic and aromatic compound. The absorbance speed from the spectra of biochar 617 1 per centimeter will be related to the OH functional group of phenol and the CCC functional group in the aromatic ring. There are characteristic analysis of cocosite. Characteristics of cocosite after being given shocking treatment with 10% and 20% concentration of bi-oil with biochar and without biochar saw observation for 15 days. The cocosite shocked in the bi-oil and covered with biochar have a low water content compared to cocoa seeds shocked in bi-oil but not covered with biochar. This indicates that biochar has an important role in the maintaining water content in seed source. Water content would affect the growth of fungi in the seed. pH value of cocosite during the storage period of 1st and to 15 days. The pH of each cocosite until the 9th day storage period was 5 until 5.5 in all treatment, except for the pH control was 6. On the 11 until 15 days of storage period, the pH for each of the cocosite increased to 5 and 5.7 until 6.1. The effectiveness of bi-oil and biochar as antifungals on cocosite. The presence of phenol, azithiazole, and hydrazine group in bi-oil and biochar of coconut shell has caused this substance could function as antifungal. The effectiveness of bi-oil 20% combined with char as fungicide on cocosite during the storage period was not indicating fungal colony found on the 10 days of observation. Well, on the 15th day, the TPC was 5.5 times 10 the power of 2 CFU per gram, and this still meets the standard national Indonesia, the maximum number of colony allowed in the seed. Conclusion: bi-oil contains a complex mixture of various chemical compounds that have fungicidal activity. Effectiveness of bi-oil 20% and biochar from coconut shell paralysis in inhibiting or killing fungal growth in the preparation of cocosite during the storage period gave the same result as shown by synthetic fungicide. Thus, bi-oil from coconut shell paralysis saw a great potential to become biofungicide. Thank you all the team for contributing to this research, the University of Halu-Oleo, and funding from the Ministry of Education, Culture, Research and Technology Kemendikbudristek, the Republic of Indonesia for the scheme of applied research. This is my presentation. Thank you for attention. Wassalamualaikum warahmatullahi wabarakatuh.
Okay. Now we have time for the discussion for 15 minutes. I think the first question is from Mrs. Maya, right? So could you please answer Pak Modi Lempang? So the question, sorry, in my chat is all deleted because I am. Bu Kanti, could you please read for the question?
Okay, I will read the question. The first question is for Mr. Modi Lempang. In all of less known hardwood samples, which one is the highest produced pulp percentage? How is the characteristic of pulp or pepper resulted, such as brightness or tear strength? And the second question is, why all those acacia species are still unutilized yet in commercially? Is it only founded in natural forest? Mr. Modi.
Okay. These all species are founded in natural tropical rainforest in South Sulawesi province. And what is the first question?
Which one, Mr. Modi? The first question. The first
Yes.
question is, in all of less known hardwood samples, which one is the highest produced pulp percentage? And how is the characteristic of pulp or paper resulted, such as brightness or tear strength?
I think I don't produce, so I don't produce the pulp. So I don't make. I just determine the yield of pulp based on its specific gravity of the wood. The highest specific gravity is simple wood species. So in this research, we don't make we don't produce a pulp. Yeah.
Okay, and the second.
Okay, so sorry, Mr. Modi. So I read your conclusion that you promote that the species you present in this presentation have a potential to use for pulp and paper industry. So that's why I want to ask what parameter that you conclude that all species can be good as a raw material for pulp and paper. I thought that it was also how do I say determine the pulp percentage. So, but if not yet, so it's okay.
Okay.
Yes.
Okay. So Pak Modi, how about the next question from Bu Maya?
Yes.
The characteristic of the pulp paper result.
He said that he didn't run
didn't produce the pulp.
Yes.
Yeah, right.
Yeah, we don't. I don't produce the pulp. So yes. So
how about the second question, Pak Modi? Why all those acacia species
only
are
found?
still?
He already answered, Pak Sukma.
Okay.
He said that only found at the tropical forest in Sulawesi, right, Pak Modi?
Yes.
So yeah. Okay,
Tropical
thank you, Pak Modi.
Natural tropical rainforest. Yeah.
Can I add a little bit a question for Mr. Modi? Mr.
Please,
Sukma?
yeah.
I just wanna know that on what age this less known wood you use in this research, because as we know that if we want to over the wood for industry, for paper industry, it has to be harvested on such a young age. But I saw that in the picture of your presentation, it has a big diameter on the wood, so I guess it has a old age or mature age rather than the juvenile one.
Yes.
So what age of trees do you use?
So I don't have I not do research about the effect of age in to produce the paper pulp, because the sample I take from natural forest. Yeah, natural forest. Yeah. Okay, so we can determine the age of the wood.
Yeah, maybe you can get information from the staff of the Ministry of the Forestry and in the field of the natural forest. Maybe Pak Modi about the age of the tree, right? Yeah, maybe next you can ask the staff.
Yeah, I think they don't know too, because in the natural not the plantation. Okay. So they don't know to the age of the tree. Okay.
Okay, Pak Modi, thank you for your answer. So the next question delivered from Ms. Fahria. Why the HHV and SP treated by 2% chitosan were demonstrate the same contact angle result? And what parameter in pulp was influenced the hydrophobicity properties? Bu ini Miss Azizatul Karimah, you will answer the question. Okay, please.
Thank you for the question, Dr. Maya. The first question is why the HSP and SP treated by 2% chitosan were demonstrated the same contact angle result. We hypothesize that in 2% chitosan concentration, doctor, the chitosan was accumulated and filled the fiber pores in the same way, both in the AHP pulp and SP pulp. So we think that could be resulting the same contact angle. And of course, this hypothesis must be proved by advanced characterization, such as morphological analysis with using FASM, for example. And this is maybe our group can do in second project of this research. And for the second question is the parameter in the pulp was influenced the hydrophobicity properties. We think that the parameter in the pulp is the existence of OH group in pulp, Dr. Maya, because we in the paper we illustrate the interaction between pulp and chitosan, which is the OH group from pulp as a hydrophilic site can interact with hydrophilic site in chitosan, so resulting the large amount of carbon ring and resulting large amount of hydrophobic site, so can resistant the water. Maybe it can answer.
Is it clear, Bu Maya?
Okay, thank you, Pak Sukma.
Okay. We still have the about 6 minutes for the discussion. So any other? Here in chat we have the question for the Bu Masuni. Yeah. The question: is there any specific consideration behind the choosing of plant-based material for lignocellulose pyrolysis for active compound? And which type of material will result in the highest yield or the most potent extract? Bu Masuni, could you please answer the question?
Yeah, I'm here.
Okay.
Yeah. I want to answer the question from Mr. Davis. For this way, I choose this material because coconut shell is dominant at Southeast Sulawesi, and then the material as the bio oil of coconut shell have material compound it is phenolic dominance. So phenolic have activity as antibacterial and antifungal.
Okay. Pak Davis, is it clear? Okay.
Yeah, thank you very much. I mean, is there any other consideration other than the material is lignocellulose-based material? For example, if we have like a plant that is very well known to have like high amount of secondary metabolites such as like I forget the class, but like tobacco or something, some type of chemical class, will it result in more potent pyrolysis result, or any type of lignocellulosic based material will result in yes, the antifungal or antibacterial extract?
This is question.
Maybe suggestion, right?
Like a follow-up question.
Yeah. Okay, thank you, Pak Davis. Okay, next question. Any other question? Oh, Bu Kanti, please.
Can I add some question for Mrs. Masuni or Miss Fitri Handayani?
Yeah, please.
Correct me if I'm wrong, if I'm not mistaken. I see that the cocoa seed is soaked into the bio oil. I just wondering for how long it's been soaked in bio oil and then cover it, or with or without the biochar. And if we want to use it in a large scale of storage of cocoa seed, can you imagine how much bio oil we'll use to soak the cocoa seed? Can we just spray it? But the problem is, if we just spray the cocoa seed, how effective it would be to prevent the what is the fungal activity or bacteria activity. And the second one is if we use bio oil to soak the cocoa seed, is it will affect the chemical compound of the cocoa seed itself, or the taste of the cocoa seed when it converts into the food or something? That's it.
The first mungkin sorry. I mean for this we shock with biochar and bio oil with for 20 minutes. And why we shock with biochar and bio oil? Because biochar have a porous and a large surface area that allows to absorb water, and bio oil have have compound as antifungal and antibacterial. And then for the second question, what is it?
When we soak it into the bio oil, is it will affect the chemical compound in the cocoa seed itself?
Oh, cocoa seed, because cocoa seed yes, usually for cocoa seed from cocoa before to the transfer with dikirim gitu to sending it for perkebunan biasanya usually pihak petani itu mengemas bijinya sebelum dikirim ke perkebunan. So kita harus, maaf ya, aku pakai bahasa Indonesia, soalnya untuk memberikan biasanya kan petani menggunakan pesticide synthetic pesticide. So we are memberikan solution for petani farmers untuk menggunakan pesticide biopesticide untuk memberikan menggantikan pestisida sintetik. Jadi biochar dapat mengurangi molekul kadar air saat saat kokosit selama storage period. Dan bioil akan memberikan perlindungan untuk menghambat kinerja activity of a bacteria or a fungal selama masa penyimpanan. Maybe
Okay, Bu. Okay, thank you, Bu Fitri ya. And maybe we can continue for the detailed discussion with Bu Kanti in the others time, in the others place, maybe yeah, because the time is over for the section 2. So we will continue for the section last section yeah in this session. Okay, thank you, Bu. Rizka, could you please show the next presenter? Just for your okay, directly. Okay, it's okay.
Okay, thank you.
So now we have the 3 last presenter for this section. So that are Bu Fala, Bu Wida, and Bu Maya Ismayanti. So here is the short bio of the Bu Faisatul Pala. She is the researcher at the Research Center for Biomass and Bioproduct, and yeah, actually she graduated from the Chemical Engineering of University Indonesia, and the current research mainly on the biomass utilization, especially on polyphenolic bioproduct and natural adhesive. And the second presenter is the Bu Wida Banar. She is also researcher at the Research Center for Biomass and Bioproduct, and she graduated from the Gajah Mada University for the master degree. And now her focus is about the nanocellulose, if I not false, right? Okay, next presenter, the last presenter in this session. Bu Maya Ismayanti. She also a researcher at the Research Center for Biomass and Bioproduct, graduated from the University of Tsukuba for the doctoral degree, and her focused research now is mainly about the tannin-based biomaterial products, yeah, include packaging, super hydrophobic, cosmetic, antioxidant, and biopesticide. Okay, let's start for the first presenter. Bu Faiz, please, your time. Play forward video.
Good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh. Thank you, moderator. Subject of my presentation today is antimicrobial activity of acacia-ammonium lignin in food packaging biofilms. This talk was conducted by me, Faizatul Falah, Denis Zulfiana, Divas Kurniawan, Dedet Sarip Nawawi, Fahria Puspitasari, Widya Fatriasari, and also Nisa Nurfarjirin Solihat. First of all, I'd like to give you an overview of this research background. Due to bacterial activity, improper usage of packaging materials might result in food contamination that can result in diarrhea, nausea, vomit, and also dizziness. Ingredients with antimicrobial qualities can hinder or prevent the growth of hazardous germs, allowing them to meet sanitary and antimicrobial standards. PLA and starch are two common raw ingredients used to produce polymers. Due to inability of starch to maintain or improve food quality, starch-based biofilm lack the potential to replace active food packaging. Therefore, it is vital to include active antibacterial substances like lignin. According to previous research, lignin derived from corn cobs as a byproduct of bioethanol production exhibits antibacterial properties. The presence of lignin's phenolic monomer inhibits enzymatic reactions and microbiological growth. This research intends to utilize lignin from pulp mill waste in the form of lignin biofilms by mixing it with glycerin as plasticizer and applying it to tapioca-based biofilm. To review, we used a single-step isolation to obtain the lignin isolate with a 6 course of washing, as can be seen here in slide, analyze its properties and antimicrobial activities, and use it in biofilm production. The methods we employed in biofilm production, namely by the dissolving method for starch lignin biofilm, Reomix for stacked PLA plasticizer lignin biofilm, as we can see here in this slide. The resulting biofilm sheets were then tested for its antimicrobial activities. Results in isolate analysis can be seen here in table 1 and 2 that presents data related to the result of the isolation of single-stage lignin from the black liquor. The result of single-stage lignin isolate yield obtained in this study was around 34.45%. As we can see here, the purity of single-step lignin is 87.69%, while the commercial lignin or sigma-aldrich lignin is 97%. Move on to the spectroscopy analysis. This figure shows the study analysis of lignin functional groups. Overall wave absorption of the research isolate was not significantly different from commercial lignin. Absorption bands at 1326 and 1213 demonstrated that single-stage lignin isolate contained single and phenolic guaiacyl group, while lignin sigma-aldrich only exhibited a 1216 absorption band, indicating only a phenolic guaiacyl group. The lignin isolate was also analyzed its thermal properties of single-stage lignin by DSC and TGA. The DSC analysis was conducted to study the process thermally occurring in lignin. Both lignin sigma and lignin single-step have two peaks in this figure. First, TG or TG1 shows the beginning of endothermic process at 25 to 100 Celsius degree when lignin water evaporates. Then the second peak, TG2, showed lignin changing from hard to pliable as the endothermic process advances, indicated by the peak point of TG2 of single-stage lignin, which was 166 Celsius degree, whereas lignin sigma peaked at 154 Celsius degree. This demonstrated the presence of required temperature differential during the transformation of lignin to plastic. And as can be seen here in TGA curve in figure 3, the weight of lignin decomposed as the temperature has increased. Lignin underwent significant degradation in 3 stages. From 25 to 100 Celsius degree is the initial stage of lignin degradation, and the second breakdown of lignin occurred between 110 to 290. And the final step of lignin degradation occurred at 300 to 750 Celsius degree, where volatile products along with lignin groups such as phenolic, alcohol, and aldehyde acids was destroyed. At this point, it can be observed that single-stage lignin group decomposed earlier than sigma lignin, which began to disintegrate at temperature of 340 and lignin sigma at 360. This is believed to be owing to the difference in lignin's raw material, with lignin sigma originating from softwood and single-stage lignin from hardwood. Let's now move forward to antimicrobial activities of the lignin isolate. Lignin isolates were dissolved using the MSO and also ethanol water at 80% concentration as a solvent, and 3 species of food-related pathogenic bacteria, that is Staphylococcus aureus, Salmonella typhi, and Escherichia coli, were used to test for antimicrobial activity. Antibacterial tests conducted on both types of lignin sample using diffusion agar method with varying concentration from 0 to 0.6 gram per milliliter demonstrated the presence of antimicrobial activity. The MSO utilized as a clean solvent as negative control here in this work had no antibacterial action against the 3 test bacteria as seen in 0% of lignin. At concentration of 0.4, antibacterial activity was greatest. This was demonstrated by the inhibition zone, which was 0.6 cm for S. aureus, 0.5 cm for E. coli, and 0.35 for S. typhi. Figure 4 here also demonstrated the result of antimicrobial test using sigma-aldrich lignin as positive control. The MSO at lignin 0% shows no inhibition zone. When lignin sigma concentration of 0.1 and 0.6 gram per milliliter exhibited greatest antibacterial efficacy against S. aureus bacteria as measured by inhibition zone of 0.25. Against E. coli, a dose of 0.5 gram per milliliter had largest inhibitory zone value. But against S. typhi bacteria, the antibacterial activity was best at 0.1, 0.3, and 0.5 gram per milliliter. with inhibition zone diameter of 0.05 centimeter. The difference in antibacterial activity between single stage and lignin sigma altric is believed to be a result of the wood raw materials utilized in the production of lignin. In this work, STV, E. coli, and S. aureus bacteria did not produce inhibitory zone to lignin ethanol water solvent containing 80% ethanol, as seen here in figure 7. This figure demonstrated inhibition zone obtained by single-stage lignin against E. coli and STV bacteria tends to be almost identical with greatest inhibitory power at concentration of 0.1 gram per milliliter and decreasing as the quantity of lignin utilized increase. The optimal dose for preventing development of S. aureus bacteria was 0.1 and 0.3 gram per milliliter with inhibition zone 0.35. Based on the result of this test, it can be stated that the concentration of 0.1 gram per milliliter of single-stage lignin inhibits the growth of 3 bacteria most effectively. At a concentration of 0.2, lignin sigma altric here prevented growth of S. aureus bacteria with inhibitory zone diameter of 0.4, and the antimicrobial activity against E. coli produced best result at a concentration of 0.1. Whereas for STV, best result was yielded at a concentration of 0.4 gram per milliliter. We also analyzed the antimicrobial activity produced using the lignin isolates. In this investigation, biofilm samples were subjected to antimicrobial testing with lignin concentration of 0, 0.5, 1%, 7.5, and 2%, which were dissolved in 80% of ethanol water solvent against S. aureus, STV, and Escherichia coli. Antimicrobial activity test on reomix biofilm with concentration of 0% as negative control did not reveal formation of inhibitory zones with a value of 0. This is because the biofilm raw material ingredients, glycerin, starch, and KLA all lack antibacterial action. And each reomix biofilm sample treated with lignin exhibited antibacterial activities as shown here, and the optimal lignin concentration for limiting the development of S. aureus was 1%, but for E. coli and STV was at 2%. Based on the finding of this test, it can be inferred that concentration of lignin antibacterial activity does not always rise in each test of bacteria. It is believed that the combination of biofilm producing component with a specific concentration of lignin affects efficiency of bacterial growth inhibition. As for biofilm with dissolving techniques, in here the absence of inhibitory zone not only indicated in concentration of 0%, in contrast to the reomix biofilm here, the dissolving technique have been demonstrated to have no antibacterial activity against S. aureus and E. coli. But against STV, the inhibited activity was great at all concentration of lignin with great success. And at 1.5% lignin concentration biofilm dissolving procedures produce best inhibitory zone diameter against STV bacteria with inhibitory zone of 0.63. I'm now approaching the end of my presentation. Allow me to summarize. The result of FTIR indicate lignin contains functional groups such as phenolic, biacyl, and seronyl. Therefore, it has implication for antimicrobial S. aureus and STV bacteria. The antimicrobial activity in lignin was optimum at a concentration of 0.1 gram per milliliter in ethanol water solvent and 0.4 in DMSO solvent. Physical and mechanical characteristic of biofilm can be enhanced through application of lignin to reomix biofilm and dissolving biofilms. In addition, both lignin-containing biofilms continue to possess antimicrobial activity. Producing reomix biofilm at concentration of 2% lignin resulted in desirable characteristic, while for dissolving techniques, biofilm resulted in 1.5% of lignin concentration used to inhibit the growth of bacteria. And to obtain optimum result in terms of characteristic and antimicrobial activity, additional analysis is required to determine the optimal combination of dissolved biofilm as well as additional tests to validate the quality of all the biofilms. In addition, it is also necessary to determine the effect of dissolving and reomix biofilm in direct contact with a food product. Thank you for listening. I hope this presentation may contribute for studies in antimicrobial activities of lignin. Assalamualaikum warahmatullahi wabarakatuh. Wish you all have a good day and delightful seminar.
Okay. Salam. Okay, next video presentation, Bu Rizka.
Assalamualaikum warahmatullahi wabarakatuh. In this occasion, I would like to present our work entitled of the effect of lignin containing nanocellulose for sugarcane leaf on agar bioplastic as an active packaging. The motivation of this research is how we can contribute to the global problem for food loss and waste because of about 33% of global food wastage come from production, storing, processing, transportation, and consumption process. And to support for the SDGs program, especially in point 12, it will be targeted for the food wastage to be declined up to 18% by 2030. Fresh consumer goods like vegetable and fruits contribute for the greatest loss than others because they continue their metabolic process after harvest. Commercial packaging, as we can see on the department store, is filmed from PP, PE, PVC, which are not biodegradable and only has an advantage on water resistance ability. And in this research, we explore one of the potential marine polysaccharide, which was agar-agar for bioplastic matrix, because it is biodegradable and compostable. Agar-agar itself extracted from red alga, namely grassland species and Galidum species, which are abundantly available in Indonesia. Furthermore, activation of bioplastic is crucial in order to extend the food shelf life and protect the food quality. The role of active packaging is to prevent gas penetration that support for metabolic process on fast food. Nanocellulose, with a diameter of less than 100 nanometers, has great better properties and modulus of elasticity, which is appropriate for active food packaging. Moreover, lignin, which has chromophore compound, has the ability as a UV absorber, considered for inhibiting the metabolic decay of the fast food product. In this research, we explore the sugarcane leaf waste for lignin containing nanocellulose because its high lignin content up to 25% and whole cellulose about 71%. And also, it also one of the potential biomass from plantation waste in Indonesia, which created about 700 million metric tons per year and hasn't abnormally utilized yet. So, the objective of this research is to isolate the lignin-containing nanocellulose or LCNF from sugarcane leaf, and also to evaluate the effect of LCNF from sugarcane leaf and plasticized agar bioplastic. This is the method and method of this research. The first one is preparation of lignin-containing nanocellulose for sugarcane leaf. We used the dried sugarcane leaf, and then we created to obtain the restrained 16 mass particles, and then the particles were alkaline pulp using 3% of sodium hydroxide, and then the pulp was processed into nanocellulose using supercolloidal Masukov grinder with rotation about 1050 and 500 ppm with several gaps micrometers for 5 cycles for each gap. And then after that, we analyze the morphological and the elemental characterization using FIS and EDS. And for the second one is the preparation of agar LCNF bioplastic. We use the 3% of agar flour and mix with the determined LCNF in various concentration in 5 up to 20% and dissolve in 150 milliliter of distilled water and stir for 35 minutes. After that, several amount of glycerol was added and continued stirring for another 20 minutes. And then the solution was cast in a metal mold, then conditioned at room temperature for 24 hours. And then continued drying in oven at 50 degrees Celsius for another 24 hours. And then the bioplastic were tested for several analyses such as physical, mechanical, and thermal properties. This is the characteristic of the LCNF from sugarcane leaf. For the chemical component, as you can see on the table, that there is a significant drop in extractive lignin and hemicellulose after treatment with respect to the amorphous part degradation, and while for the cellulose content rise by about 41% as an exposed crystalline part. And for the LCNF characterization, as we can see over here, that the suspension result in bony and stable suspension. And well, for the side micrograph reveals for the fibrillars and filament form with a relatively high aspect ratio, which corresponds to the cellulose nanofibrils. From the side micrograph, we can see that some white spot identified covering the fibers. And by the EDS, we identified that there is a silica element in about 13%. This is the characteristic of agar LCNF plastic in physical properties. As we can see over here in the table, the thickness of agar LCNF bioplastic is similar to the plastic agar, ranging from 0.1 up to 0.12 millimeters. And the moisture content of bioplastic is higher than plastic agar and tend to be increased by the LCNF addition. Well, for the right one is the water vapor penetration, is the ability of bioplastic to restrain water vapor. From the VVP of agar LCNF bioplastic indicates a gradual decrease by the addition of LCNF in a various composition, which means for the improvement of barrier properties. The LCNF up to 20% could improve the BVP by about 73%. So this is the water resistance analysis, including water uptake ratio and water solubility. The bioplastic have a lower water uptake ratio than plastic agar and tend to be lower by the addition of LCNF. LCNF addition up to 20% could improve the water uptake ratio by about 54%. And water solubility has the same result as water uptake ratio, with the addition of LCNF also decrease the water solubility. That confirmed for the LCNF containing lignin, also incorporated with silica dioxide, may provide better resistance to water for agar bioplastic. And this is the UV block and transparency analysis. The existence of lignin produces browny and darker bioplastics and affect its transparency, which could block the UV wave exposure. From the table, we can see, especially for the transmittance at 2080 and 400 nanometers, are prone to be declined as the LCNF addition. And the transparency of bioplastic, which observed at 660 nanometers, the transmitted are declined by the addition of LCNF, indicate for unclear bioplastic product but remains transparent. This is the mechanical properties of the bioplastic. The elongation at break starts to increase in 10% of LCNF, then drops in 20% of LCNF. And optimum elongation at break results in 15% of LCNF in about 22% than plastic agar. In another hand, tensile strength tends to be decreased as LCNF content increase in about 38% and for modulus of elasticity in about 69% for LCNF up to 20%. This is the thermal properties of the bioplastic performed by DST and PG analysis. From the DST analysis, informed that the melting temperature of bioplastic tends to slightly decrease by increasing of LCNF content. This may influence by moisture content that affect for the overall molecular structure of agar bioplastics. And for the TGA analysis, informed that the LCNF have 2 stages of decomposition, while bioplastic have 3 stages of decomposition. From the table, we also can see that the LCNF has high onset temperature in about 300 something degrees Celsius and maximum decomposition temperature about 342 degrees Celsius. That because the lignin content also silica dioxide, which more thermally stable than other carbohydrates like cellulose and agar. And when LCNF are subjected to the agar bioplastic in 5 up to 20%, the onset temperature reduces compared to the plasticized agar, partially in stage 2. But at stage 3, the onset temperature becomes higher than plasticized agar. However, the increasing LCNF content enhances the thermal stability, reviewed from a maximum decomposition temperature and weight loss for agar and LCNF bioplastic. By the result, we can conclude the first one is we can produce the LCNF with high aspect ratio and fibrous filament by ultrafine binder in several cycles in various zone gaps. And the study also giving a novel finding of LCNF incorporated with residual silica dioxide of sugarcane leaf. And the second one is the LCNF incorporated with silica dioxide residue have positively affect for water resistance ability, but negatively affect for UV blocking ability. And the mechanical behavior rather decrease, especially for tensile strength, but improve for elongation at break. And the thermal stability of agar LCNF plastic improve gradually as LCNF loading. The LCNF remarkably active as a gas barrier for food packaging. Okay, this is the end of my presentation. Thank you for your kind attention.
Rizka, last video presentation from Bu Maya.
Good afternoon, ladies and gentlemen. It is an honor for me to be able participating in the second international conference of Lignocellulose. In this opportunity, I would like to share my research that was a collaboration between BRIN with the Research Center Chemical and Packaging, Ministry of Industry, and also some college from the University of Lampung. And it was also part of the Justice project with Tobimatsu Sensei from Kyoto University, Japan. The title in here is Optimization of Tannin Isolation from Bark of Acacia Mangium and its application in PLA tannin-based biofilm with antioxidant properties. As an introduction, we know that during COVID pandemic and even after that, post-pandemic, there was a lifestyle changing. People more like in using food delivery application in order to prevent contamination of virus and also just for a convenience perception. So what was that kind of it? It was increase the utilization of single use with packaging that most are non-biodegradable. So that agency to packaging, good freshness of the grade. One important is the food packaging is using and use as the recycled material. So in here we from pulp and paper in as a precursor or building block, phenol or aromatic as antioxidant. Objective of this research optimization condition for tannin extraction from bark of Acacia mangium, and also to synthesize the problem of PLA tannin base with antioxidant activity and also their characterization. Next, we move to the material and method. In here, the first we need to isolate the tannin from bark of Acacia mangium, where is in the previous study, we did also the tannin extraction from Acacia crassicarpa and Acacia meranti using 70% acetone. But unfortunately, it was less applicable on the large scale. So in here, we try to use another solvent with similar polarity with 70% acetone. We use water with various temperature from 60 to 100 degrees Celsius. We know that water itself is a polar solvent, but when we increase the temperature, so the polarity it will be decreased. So hopefully we can get a condition optimum at the temperature variable that can reach the same polarity as the 70% acetone and get tannin compound from the bark of Acacia mangium. Next, after we get the bark extract, we need to do assessment for antioxidant properties and also content with pH test. And also for quantitative phenolic, we use Folin Biocalc as a reagent. Next, here, as you see that in table 1, we have the yield percentage, content and also the antioxidant activity. Along represented with in degrees Celsius. U is presented by 80 degrees Celsius. Sorry, 90 degrees Celsius with the percentage about 26.59%. But unfortunately, it slightly decreased at 100 degrees Celsius with the total yield about 25%. On the contrary, the phenolic compound it was decreased in increasing water temperature, and the highest content of phenolic in here seems to have a positive effect on the antioxidant activity. At 60 degrees Celsius with the highest GAA or phenolic compound, 45 milligram per gram, show the highest antioxidant activity that was reported with the smallest AC50 is about 14.60 microgram per milliliter. By this result, we have one question: why the highest randomized by increasing water temperature are not follow with the highest phenolic content? So we want to know what was exact the component extracted or isolated from the bark. So here, as an additional data, we have pyrogram by PYD-CMS analysis to do the rapid screening for tannin, lignin, and also carbohydrate derivative. The table next to pyrogram, in here we can see that the lignin is quite similar along the variation of temperature. It's about 8%. And in case of tannin, from 60 to 90, there was, how do I say, almost same or tend to increase. But after that, the tannin content is decreased until 100 degrees Celsius that only get 57%. And as opposite with the tannin, the carbohydrate, as you can see that it's tend to increase by increasing the temperature. So from here, we can get a conclusion that the antioxidant activity is related to the phenolic compound, but not only tannin, the lignin content also have effect into the activity. And in the opposite side, the carbohydrate have influence in decreasing the antioxidant activity. As you can see that at 100 degrees Celsius, the total phenolic is how do I say, smaller compared to the 60 or 70, and also they have a higher EC50 due to the high amount of carbohydrates derivative. Next, confirm the bark extract have the antioxidant activity. So the next method is we do formulation of PLA tannin biofilm. In here, we do the two kind of method, casting and extrusion. The A is a casting, then extrusion. For casting, we use acetone as a solvent. While in the event, just put tannin. After that, we do pressing. We get the biofilm, and after we get the biofilm, we continue for antioxidant activity again, and also some analysis for characterization. And here, this is the result for PLA tannin-based biofilm composite. The figure 1, as you can see, as the visual appearance, find that the biofilm made by extrusion is darker than made by the casting. Here you can see that CF is darker compared to BC, and also we can see that the solubility by extrusion method is better compared to the casting method. From this result also we can get a conclusion that the solubility problem might cause this phenomena, and the extrusion method produce a good dispersion tannin in the PLA matrix. Okay, after that the biofilm we do for antioxidant activity in here, AA percentage. As you can see that, how do I say, both method, I mean like casting and also PLA, when we add the tannin and also citric acid, there is increasing of antioxidant activity. For number C, casting PLA-TCA, they have 66%, and for extrusion method, they have a highest one as the 92%. Another physical property that was conducted is the contact angle. As you can see that all the biofilm have the super hydrophobic with the higher theta C that were more than 150 degree. And in case of the mechanical properties, we have young modulus, tensile, and elongation. In both young modulus and tensile, the aiding treatment by tannin and citric acid, it would decrease the value. And for elongation break, the tannin treatment can be increased, and for citric acid, it was decreasing the elongation at break percentage. As a conclusion, we have 3 points. The first, the crude tannin extract of bark of Acacia mangium have a potency oxidant in food packaging, and as optimum condition of isolation is using water with the temperature 70 degrees Celsius. And from both method, casting and extrusion, PLA tannin have antioxidant activity by decreasing the free radical scavenger with the value about 65% and 86%. But unfortunately, the mechanical properties of the biofilm was stiff, and I think further investigation needed to improve by adding plasticizer. As acknowledgement, us give the full acknowledgement for funding from research program or DIPA Rumah program, and also for Justice Risk grant, Kyoto University, and also iLab BRIN for the facility. That's all my presentation. Thank you very much. We are
Okay, because of the limited time, we just have maybe one question. Bukanti, you have question?
How do you know that, Mr. I want to ask Mrs. Maya.
Okay.
Oh, there's a question also from Mr. Davis in the chat
Oh,
room.
yeah, okay, maybe priority is from
Yes.
the participants,
Okay,
so please
okay.
read. Ah, yeah, also same deliver for Dr. Maya. I was wondering if tannin content is maintained in decaying plant material, such as wilting leaf. Does plant material have to be fresh for maximum extraction? Bu Maya?
have to be fresh. Oh, sorry, I'm not. Does
Such
plant
clear question?
material? What do you mean with fresh from the maximum extraction? Actually, we
The fresh
get
material, Maya, Bu Maya. Maybe the meaning of the question is fresh material, yeah.
do
So,
I use a fresh material? You mean?
Yes, yes, I think.
Okay. Oh, okay. Yeah. Actually, since the main goal, main objective of this research is one to enhance the utilization of byproduct coming from the pulp and paper industry. So we use how do I say, the real acacia chips bark chips coming from the pulp and paper industry. That's due to the high, how do I say, humidity. Some of the bark chip, it was decay. So that's why in the first we got from the pulp and paper industry, we need to wash and also dry from 2 until 3 days to remove the decay before we continue for the extraction. So maybe I just can say like that. So Pak Davis.
Pak Davis, is it clear from?
Thank you very much. I want to know if the plant is not fresh, the tannin content is still there, right?
Yes, still there.
Okay,
Yeah.
thank you very much.
Yeah. Actually, tannin also have anti-decay activity, but maybe due to the high humidity in plant area, usually wet or maybe a bit, how do I say, contain water. So that's why the sample it's how do I say, not really good enough, or the humidity is too high. Yes.
Thank you very much.
Yeah, thank you.
Okay. Because of the time, I think we have to end of this session. So now, before I close the session, I will inform to all of you about the best presenter and the best authors will be announced in the second day of this conference in the ceremony, closing ceremony, right, Bu Kanti? So please still have time for attending the conference in the second day to know who is the best presenter and best author. So I think I am sure we still have the many questions and curiosity of our presentation for this presentation. So because of the time, we have to close in this first session in conference today. So I would like to thank to all of the presenters for an interactive session and in such a joyful presentation today. And we will continue tomorrow on the second day of the second ICONLIG 2022. So please still attend for the tomorrow's session. I think the question will be more in the next session. Thank you for your presentation. We are looking forward to see you tomorrow and have a great race for you today. And before we close the session, please open your camera to take the picture. Okay, Bu Rizka, please help us for the picture. Pak Andrian, still have. Do you still there, Pak Andrian, Bu Is, Bu Devi, Bu Riki? Before we take a picture,
I'm using OpenCore system.
Please open your camera. Okay, please wait, Bu Rizka, please wait. Bu Devi, Bu Riki, Bu Muslimah, Pak Esther, could you please open the camera? Okay, Bu Rizka, let's start for take a picture.
Okay.
So please, please.
1, 2, 3. Smile. And the second page. 1, 2, 3. Please smile. Okay, finish. Thank you.
Okay, thank you for all participants. We will close our session today. Have a nice day. See you tomorrow.
Thank you very much.
Thank you, everyone.
Thank you, everyone.
Thank you, Chair, Vice Chair. Thank you, everyone.
See you next.
Recording stopped.